Welcome back. Now we are entering the first session on topic cashew nut shell liquid and its derivative in healthcare. I would like to invite Dr. Dinesh Kumar PM, Professor and HOD, KMCT Ayurveda Medical College, as a chairperson of the session. Dr. Dinesh Kumar PM, who has more than 20 years of experience in academic, he has also published various articles in national and international journals. He also presented scientific papers in esteemed conferences. He has also organized and participated in many workshops in his discipline. I invite you, sir, for chairing the session.
നമ്മുടെ ചെയർമാൻ ഇനാഗുറൽ സെഷനിൽ പറഞ്ഞൊരു കാര്യമുണ്ട്. അതായത് എല്ലാ ജീവജാലങ്ങളെയും നമ്മളെപ്പോലെ കാണണം എന്നുള്ളത്. ആയുർവേദത്തിൽ ആചാര്യന്മാർ പ്രത്യേകം പറഞ്ഞിട്ടുണ്ട്, ആത്മവത് സതതം പശ്യത് അപ്പി കീട പിപീലികാം. അതായത് നമ്മളെപ്പോലെ ഈ ഭൂമിയിലുള്ള എല്ലാ ജീവജാലങ്ങളെയും, അതായത് ചെറിയ പുഴുക്കളും എറുമ്പുകളെപ്പോലും നമ്മളെപ്പോലെ നമ്മുടെ ആത്മാവിൻ്റെ അംശം പോലെ തന്നെ കാണണമെന്ന്. അതേ കാഴ്ചപ്പാട് തന്നെയാണ് നമ്മുടെ ചെയർമാനുള്ളതെന്ന് കേട്ടപ്പോൾ എനിക്ക് വളരെ സന്തോഷം തോന്നി. നമ്മുടെ ഈ KMCT ഇൻസ്റ്റിറ്റ്യൂഷൻസിൻ്റെ പ്രത്യേകത എന്തെന്ന് പറഞ്ഞാൽ എല്ലാ ഡിസിപ്ലിൻസും ഒരു കുടക്കീഴിൽ അവൈലബിൾ ആണെന്നുള്ളതാണ് ഇവിടുത്തെ പ്രത്യേകത. അതുകൊണ്ട് തന്നെ നമുക്ക് എല്ലാത്തരം ഗവേഷണങ്ങളും nowadays it is management is showing much interest in interdisciplinary researches. എല്ലാ ആയുർവേദമായാലും മോഡേൺ ആയാലും ഡെൻ്റൽ ആയാലും എല്ലാത്തിനെയും ഉൾക്കൊള്ളിച്ചു കൊണ്ടുള്ള ഒരു ഇൻ്റർഡിസിപ്ലിനറി റിസർച്ചിന് വളരെയധികം പ്രാധാന്യം KMCT ഇപ്പോൾ കൊടുക്കുന്നുണ്ട്. അത് വളരെ നല്ല കാര്യമാണ്. കാരണം ഇന്ന് അടുത്ത കാലഘട്ടം ആവശ്യപ്പെടുന്നതും അതുതന്നെയാണ്. നമുക്കറിയാം, മോഡേൺ മെഡിസിനിൽ പ്രധാനമായിട്ടും കെമിക്കൽ ബേസ്ഡ് ആയിട്ടുള്ള മെഡിസിൻസാണ് ഉപയോഗിക്കുന്നത്. ഞങ്ങൾ ആയുർവേദത്തിൽ പ്ലാന്റ് ഒറിജിൻ ആയിട്ടുള്ള മെഡിസിൻസാണ് ഉപയോഗിക്കുന്നത്. അപ്പോൾ ഇതിൻ്റെ ആയുർവേദത്തിൻ്റെ ഒരു കൺസെപ്റ്റ് എന്ന് പറഞ്ഞാൽ അത് ശരിക്കും പറഞ്ഞാൽ ഒരു ബയോ ഫിസിക്സാണ്. അതിന് കെമിസ്ട്രിയോടല്ല കൂടുതൽ അടുപ്പം, ഫിസിക്സിനോടാണ്. നമ്മുടെ ആയുർവേദത്തിൻ്റെ അപ്രോച്ചുകളൊക്കെ ഒരു ഫിസിക്സ് റിലേറ്റഡ് ആയിട്ടുള്ള, അതായത് ഗുരുത്വം ലഘുത്വം എന്നൊക്കെ പറഞ്ഞുകൊണ്ട് ആ ഒരു അപ്രോച്ചിലാണ് ചികിത്സ സിദ്ധാന്തങ്ങൾ അതിൽ ഉൾക്കൊള്ളിച്ചിട്ടുള്ളത്. അതുകൊണ്ട് തന്നെ ഈ കെമിസ്ട്രി ബേസ്ഡ് ആയിട്ടുള്ള ഒരു ശാസ്ത്രത്തിൻ്റെ സിദ്ധാന്തങ്ങളിലേക്ക് അതിൻ്റെ ശ്രദ്ധ പോകാത്തത് കൊണ്ട് തന്നെ ധാരാളം പഠനങ്ങൾ അതിൽ നടക്കേണ്ടതായിട്ടുണ്ട്. ആയുർവേദത്തിൽ നടക്കുന്ന പഠനങ്ങൾ എന്ന് പറയുന്ന 1% പോലും ആകുന്നില്ല എന്നുള്ളതാണ് സത്യം. ഒരുപാട് ഗവേഷണങ്ങൾ നടക്കേണ്ട ഒരു വിഭാഗമാണ് ആയുർവേദം. അതുകൊണ്ട് തന്നെ ഇത്തരം ഇൻ്റർഡിസിപ്ലിനറി ആയിട്ടുള്ള, ഇവിടെ എല്ലാം മലയാളം അറിയുന്നവരാണെന്ന് വിചാരിക്കുന്നത് അല്ലേ? യെസ്. ഇൻ്റർഡിസിപ്ലിനറി ആയിട്ടുള്ള ശാസ്ത്രങ്ങളുടെ ഒരു യോജിപ്പ് അത്യാവശ്യമാണ് ഇനി വരുന്ന സമൂഹത്തിൻ്റെ ആരോഗ്യപരമായ വളർച്ചയ്ക്ക്. അത് അത്യാവശ്യമാണ്. ലോകം മുഴുവൻ അതിനുവേണ്ടി ശ്രമിച്ചുകൊണ്ടിരിക്കുന്നു, നമ്മളും അതിനുവേണ്ടി ശ്രമിച്ചുകൊണ്ടിരിക്കുകയാണ്. നമുക്കിന്ന് ഇവിടെ 3 ദിവസമായിട്ട് നടക്കാൻ പോകുന്ന ഒരു ഹെൽത്ത് റിലേറ്റഡ് ഒരു ഫീസ്റ്റാണ് നടക്കുന്നത്. അത് അത്രയും എമിനൻ്റായ ഫാക്കൽറ്റീസിനെയാണ് നമുക്കിവിടെ കിട്ടിയിരിക്കുന്നത്. അതിൽ ഇന്ന് നമുക്ക് കിട്ടിയിരിക്കുന്ന ഫാക്കൽറ്റി ഡോക്ടർ KI സുരേഷ് ആണ്. അദ്ദേഹം CSIRൻ്റെ കീഴിലുള്ള നാഷണൽ ഇൻസ്റ്റിറ്റ്യൂട്ട് ഫോർ ഇൻ്റർഡിസിപ്ലിനറി സയൻസ് ആൻഡ് ടെക്നോളജി അതിൽ ചീഫ് സയൻറ്റിസ്റ്റാണ്. മെറ്റീരിയൽ സയൻസ് ആൻഡ് ടെക്നോളജി ഡിവിഷനിലെ സയൻറ്റിസ്റ്റാണ് അദ്ദേഹം. BSc കെമിസ്ട്രിയിൽ BSc പാസ്സായിരിക്കുന്നത് MG യൂണിവേഴ്സിറ്റിയിൽ നിന്നാണ്. അതും അവിടുത്തെ ഏറ്റവും കൂടുതൽ മാർക്ക് കിട്ടിയ സ്റ്റുഡൻ്റായിട്ടാണ് അദ്ദേഹം പുറത്തിറങ്ങിയത്. അതിനുശേഷം MSc അവിടെ നിന്ന് തന്നെ MG യൂണിവേഴ്സിറ്റിയിൽ നിന്ന് എടുത്തു. പോളിമർ ടെക്നോളജിയിലായിരുന്നു. അതിൽ ഫസ്റ്റ് റാങ്ക് അദ്ദേഹത്തിന് കിട്ടി, യൂണിവേഴ്സിറ്റി ഫസ്റ്റ് റാങ്ക്. അതിനുശേഷം PhD ഇൻ അപ്ലൈഡ് കെമിസ്ട്രിയിൽ PhD IIT
So thank you, sir, for your kind words of introduction. So I will try to do justice, because I am not a person of pharmaceutical chemistry or science background. Basically, I have done my, as he has already mentioned, I have done my graduation in chemistry. Then I studied polymer chemistry, then polymer plastics and rubber technology. And in my profession, we do all kinds of interdisciplinary research, starting from industrial chemicals to health, surfactant science, and all. So before I start with, I wanted to give a brief introduction to my organization, that is the CSIR, and CSIR stands for Council of Scientific and Industrial Research. So there is no medical science involved in that. And then I belong to the National Institute for Interdisciplinary Science and Technology, that is it was previously the Regional Research Laboratory, that was renamed in the around 2010 as NIST. So we have basically 6 R&D departments. I belong to the Material Science and Technology division. Then there is a Chemical Science and Technology division where they also work on pharmaceutical chemistry. And then there is a Microbial Science and Process Technology. Then there is an Agro Processing Technology division, and then Environmental Technology division. So all these departments are very important for societal developing technologies and various interventions for societal development. So you can get more information about our institute as well as the program. We don't have any undergraduate or master's program. We only have PhD students and project assistants whom we take for carrying out the project work. And altogether, there are around 37 laboratories throughout India, CSIR laboratories. The one in Kerala is in Trivandrum. So if you look at my research profile, so basically I do research in the area of chemistry and material science, where involved with the polymer synthesis, then product development, then structure property correlation studies to understand the performance of the material, and the sustainable chemistry with renewable feedstocks. So coming to my talk I wanted to give an overview of cashew nut shell liquid and its components for pharmaceutical applications, basically because I have been working with the cashew nut shell liquid, that is a renewable resource material for various kinds of industrial chemicals, paints, coatings. But during my research, I have also found that it has got very important applications in the pharmaceutical area also. And luckily, I have also got a student who had interest in developing some pharmaceutical derivatives based on this material. So if you look at drug design and discovery, you know that it is a very long process, and bringing a new drug molecule or a pharmaceutical to the market requires more than a decade, about 10 years. So and there is an interest for developing natural products, because Ayurveda and all, if you look at it, consists of various natural products, and for various reasons, because of its availability, cost considerations, or less side effects, they have all become important, and we can create compound libraries to understand their performance, and then new techniques like computational chemistry approaches can be applied for the development of newer bioactive molecules. So if you look at the need for developing new drugs or pharmaceutical, the most important are the treating bacterial and viral infections. They require using new therapeutic approaches to overcome drug resistant diseases. So we have seen the very clear example is the recent times we have the COVID breakout, and you have all experienced how difficult it was to treat with the available medicines or systems. So natural products are identified as important research for drug discovery and yield potentially powerful therapeutic agents. So the cashew tree, which you all may be familiar, is originally a native of Brazil, but is cultivated now throughout the world. And chemically, it contains a compound called anacardic acid, which is a convenient salicylic acid system, because acetyl salicylic acid is commercially aspirin. You know that it is useful in various kinds of inflammation and pain. It's a non-steroidal molecule. So this cashew nut shell liquid contains this anacardic acid, that is a can be considered as an analog of salicylic acid, and it is also carrying a side chain with varying degree of unsaturation. So this cashew nut shell liquid is obtained from the plant species cashew nut Anacardium occidentale, and the shell oil is extracted from, because the cashew kernel we take it for edible purposes, and there is an oil present in this outer shell material, that is the cashew nut shell liquid. And it has been, as I have mentioned, it has been used in the various industrial applications like paints and varnish, then polyurethane polymers, surfactants and epoxy resins, and in medicine and pharmaceutical industry it has given rise to the development of chemical intermediates, drug delivery systems, because of the presence of this phenol nuclei and an unsaturated side chain. It may form vesicle type of systems which are important for drug delivery applications, and then lead compounds to treat various diseases, antibacterial and antiviral conditions. But if you say cashew nut shell liquid, it is not a simple compound. Being a natural product, it has got varying composition, and the method how you extract the oil from the shells of the cashew nut is very important. So there are basically two processes that is used: the cold process, which is by using chemical solvents, or you can apply heat and extract by pressing the shells. So they are known by different names because of the composition. So only this oil extracted by the cold process or solvent process will have anacardic acid as the major component, and then there are other components like cardol, cardanol and some other minor components. Whereas the oil that is extracted by heat treatment contains the major component is cardanol, and but it doesn't contain any anacardic acid, which is the biologically important molecule. So this is the structure of cardanol, and this is anacardic acid. So this is what we have mentioned it as. It is an analog of salicylic acid, but with a alkyl chain in the meta position of the benzene ring. So this structural combination actually offers varying opportunities for the synthetic chemist, medicinal chemist or material scientist to develop various kinds of products. So we can carry out the process like esterification, epoxidation or ethoxylation reactions on this phenolic OH, or this unsaturated side chain we can carry out hydrogenation, then epoxidation, and make an adduct with phenols, and then we can also carry out the simple reactions like nitration, sulfonation, bromination for this benzene nuclei, which are all yield biologically different active compounds. So as I mentioned, the structure of anacardic acid is characterized by salicylic acid structure with a 15 carbon side chain of varying unsaturation. It is a potential lead compound for neutrophil function in innate immune defense of the living systems. So these are the chemical structures. This is anacardic acid. It's a phenol with an unsaturated side chain as well as a carboxyl group, whereas cardanol contains phenol with only a alkyl side chain, and then the cardol we will have two hydroxyl groups, and then there is a methyl substituent in this diphenol if it is 2-methyl cardol, and this is the varying composition of this unsaturation, the side chain. There is a monoene, there is one double bond, two double bonds, and three double bonded systems are present. So accordingly, so all this requires a chemist's expertise to analyze, understand the structure, and developing further methodologies for its modification. So it has been the only anacardic acid has been reported or used in the literature to develop various biologically active compounds for antibacterial, anti-inflammatory and anti-cancer agents. So cancer is still one of the major diseases causing death worldwide, and there is an interest for developing anti-cancerous derivatives of this anacardic acid based systems. So it has been found that anacardic acid kills directly gram-positive bacteria, and when anacardic acid is added to freshly isolated human neutrophils, they promote this is the mechanism by which it was found to be biologically active. They create the formation of reactive oxygen species and neutrophil extracellular traps, the mechanisms used by neutrophils to kill an invading bacteria. So the alkyl chain plays a key role in activation of the neutrophil oxidative burst as confirmed by model studies using. So to confirm how you can establish that this anacardic acid present in cashew nut shell liquid has a better activity. So some researchers have carried out studies using salicylic acid, which doesn't have a saturated or unsaturated side chain, but the phenol and carboxyl groups are there. So through these studies, such properties were examined. So this is some derivatives of anacardic acid, the sulfonamide derivatives, then sildenafil, which are used in treatment of sexual diseases, and then macrolactones. These were all examples of compounds earlier developed using this anacardic acid. And we have also developed some derivatives through functionalization of the phenolic OH groups to develop the polyols, which is a nitrogen containing heterocyclic compound, and then some amino derivatives and then quaternary ammonium compounds. These compounds may be important in the formation of lipid delivery vesicles for delivery of various drugs. So it has also been used to develop hybrid systems. For example, you combine this natural phenol with some other materials like porphyrin, which is a carbon nanomaterial, then fullerenes, these are also carbon nanomaterials. So they will have different activity when you hybridize with this kind of molecules. So we have studied the synthesis of some new heterocyclic compounds, that is the benzoxazine derivatives, through the one-pot Manich reactions, where they were found to be useful in the treatment of bacterial infections and fungicidal and anti-inflammatory disease conditions. So I don't know why. So as I mentioned, the presence of the anacardic acid in the cashew nut shell oil depends upon the extraction method used. So we have to, first of all, for example, for biological or pharmaceutical application, it has to be first purified to the pure form of anacardic acid. But commercially this anacardic acid is not available, and also if it is available from some multinational companies, but they are all very extensively priced. A few milligrams will cost 50,000, 1 lakh rupees, in that range. So we have developed a methodology for extraction of this anacardic acid, starting from cashew nut shell liquid, the crude oil that is extracted from the shell by the cold process. If you take, and you can develop methodology using chemicals like calcium oxide, you extract it as a calcium salt and then purify it so that you will get a pure form of anacardic acid, and then it is further purified by solvent evaporation. So the difficulty with the extraction of this anacardic acid from the natural oil is that if you heat, it will decarboxylate, the carbon dioxide will be eliminated, and you get a simple phenol that is cardinol. That is the most common form of. So for the benzoxazine synthesis, this is the chemistry we have used. You take a diamine and then the cardinol and then formaldehyde. You can develop a benzoxazine type of system. So this, and then we carry out the structural characterization by various techniques like nuclear magnetic resonance, you might have all heard, where it will indicate the presence of different type of protons in the compound, and also the 13C NMR, which will give characteristic peaks for each type of carbon present in the molecule. So this is how we characterize once we do some synthesis of a new molecule. How we characterize, we take the spectral details, and then characterize. Then we also take the mass spectroscopy, where the molar mass of the molecule can be found out. Then IR spectroscopy, all these, a combination of techniques will be used for its structural characterization. And then the purity will also be checked by high performance liquid chromatography, HPLC, where the compound will be passed through a column using a solvent, and depending upon the number of components present, it will also indicate the presence of any impurities present in the compound. So this is again, we have also, this is again based on, we have prepared the benzoxazine derivatives starting from anacardic acid, cardinol, as well as hydrogenated cardinol. So all these compounds will be potentially useful biologically active compounds. So we have, our objective was to make a comparative study on the effect of the side chain unsaturation or the anacardic acid presence or a cardinal structure on the biological activity. So these are all, and again we have varied the amine type that is used in the preparation of the benzoxazine, and its structure was also characterized by HPLC and other techniques. So this is some of you may be interested to know more about the biological application and also about cashew nut shell liquid. So this literature presented here, there are biologically important journals. The recent literature or the background literature is normally published in journal articles. So you can refer to some of these publications. So, and we have also evaluated some preliminary studies on the anti-cancer activity studies, which I have not presented here because they are yet to be published. And they have shown moderate to good activity, and where the presence of the side chain unsaturation was revealed. So that helps in biologically activity of the molecule. That is, so this is apart from, as I mentioned, so I have started with working with cashew nut shell liquid, various kinds of derivatives, not only for biological application but mainly for industrial application. But during the research it was found out about this biological activity, and one pharmaceutical student only was interested to take it up for their dissertation work, and that is how that work was executed. And and we have also developed some fluorescent compounds based on these cashew nut shell liquid derivatives, which are important for imaging applications or other applications, and imaging or security application, for example to develop inks for security application. So they may be important. So this is the current research activities are based on developing carbon dot materials, which is again important for biological applications like controlled delivery systems, and then composite systems for inks, and then polyurethane dispersions. By developing polyurethane dispersion based on this natural oil, we can get better sustainable approaches to develop many of the industrially useful materials. And this is what I mentioned about the carbon nanomaterial. So the carbon dot work is already going on, and then because graphene, you might have heard about graphene, is a material, carbon nanomaterial that has been awarded the Nobel Prize in 2010, and that is projected to be a material of outstanding properties for various application that is troubling the human. For example, in biomedical application, energy, alternative energy systems, battery storage systems, renewable energy, all this it is important. So we have also developed some carbon nanomaterial graphene based on the literature reports, and we have studied in biomedical application, we have studied its pro-angiogenic behavior. Angiogenesis is the formation of new blood vessels, and this carbon nanomaterial, when used in nanogram concentration, it has been found to give promote the growth of blood vessels. So they may be useful in developing coating systems for some implants like stents and all, where such properties may be preferable. And we have also developed some surfactant type of systems. So this, if you look at This is a graphene sheet but with polymer chains attached to the surface, and you can control the molecular weight of this polymer as well as the type of function groups present on this polymer, so that it may form micellar type of systems which are important for drug delivery applications. So we have also studied this nanocomposite performance in sensing application, electrochemical sensing of dopamine and ascorbic acid. These are biologically important molecules, and so its concentration determination may be important in various products like fruit juices and all. So this is another part of the work. So this is now I have come to the end of my presentation. So I would like to thank the director and NIST for the permission, the various funding agencies, industries for the sponsored work, as well as my prior organization, IICT Hyderabad, and then CSIR NIST in Trivandrum, as well as my collaborators from Germany. So I thank you for your attention.
Thank you, sir, for sharing your knowledge with us. Now
I'll be happy to answer any queries if you have.
Thank you, sir, for a wonderful presentation. We had this presentation basically on it's a molecular level of the cashew nut oil, and its recent techniques to extract it. And ഞങ്ങൾ കഴിഞ്ഞ ദിവസം സാറിന്, അതായത് നമ്മുടെ ഫാർമസി കോളേജിൽ പ്രിൻസിപ്പളിന് ഒരു റിസർച്ചിൻ്റെ ഒരു ആർട്ടിക്കിൾ അയച്ചു കൊടുത്തിട്ടുണ്ട്. അതിൽ സീറോസിസ് എന്ന് പറയുന്ന രോഗത്തിൽ ഈ കാഷ്യൂനട്ട് ഓയിലിൻ്റെ ഒരു യൂസിനെ പറ്റിയുള്ള ഒരു ആർട്ടിക്കിളാണ്. അത് പബ്ലിഷ് ചെയ്യപ്പെട്ടിട്ടുള്ളതാണ്. വളരെ സാറ്റിസ്ഫാക്ടറി ആയിട്ടുള്ള റിസൾട്ടാണ് അതിൽ കിട്ടിയിട്ടുള്ളത്. അപ്പോൾ ഇത്തരം രോഗാവസ്ഥകളിൽ ഈ കാഷ്യൂനട്ട് ഓയിൽ നമ്മൾ അതിനൊരു സ്റ്റഡി കൂടെ നടത്തണമെന്ന് ഇവിടെ ഫാർമസി കോളേജുമായിട്ട് ചേർന്നിട്ട് നടത്താൻ തീരുമാനിച്ചത്. അപ്പം അതിൽ വന്നൊരു ഡൗട്ട്, ഇതെങ്ങനെ എക്സ്ട്രാക്ട് ചെയ്തെടുക്കും എന്നുള്ളതായിരുന്നു. അപ്പോൾ അതിനുള്ള വിദ്യയാണ് സാറ് ഇപ്പം വളരെ ചിലവ് കുറഞ്ഞ രീതിയിൽ അത് എക്സ്ട്രാക്ട് ചെയ്തെടുക്കാനുള്ള ടെക്നോളജിയും വിദ്യകളുമാണ് ഇപ്പം സാറ് പറഞ്ഞു തന്നിരിക്കുന്നത്. അതുകൊണ്ട് തന്നെ വളരെ യൂസ്ഫുള്ളായൊരു ക്ലാസ്സായി മാറിയിരിക്കുന്നു ഞങ്ങളെ സംബന്ധിച്ചിടത്തോളം ഇത്.
Yeah, this is as if you look at from my viewpoint, I was only interested in how to extract this. I don't have any expertise in the pharmacological evaluation. If you people with your expertise come up, then you can do much better work that is really useful to the society. You can, maybe if somebody wants, they can start an industry itself, because as I mentioned, the only company that is supplying anacardic acid is now Sigma Elrich, and they charge around 50,000 rupees for 50 milligram of the compound. There have been inquiries from US and other European companies also for these derivatives.
So that's why it is a very apt presentation for this session. And any doubts we can clear here. It is open for you.
I tried to make it as simple as possible, so only the fundamental things were, no hi-fi science was presented.
Yes.
Yes.
Sir, it was a wonderful presentation.
Thank you, sir.
It's a wonderful presentation. And the question is that anacardic acid, and basically that is salicylic acid molecule.
Yeah.
And as you have already mentioned, aspirin is a wonder drug. Right from its discovery, still it stands in the market. So I hope that this molecule can make some wonders
Yes.
in the treatments. And you have mentioned that that result is not published. Which type of cancer, for which type of cancer that is effective?
That is we have tested against HeLa cell lines and some
HeLa cell lines.
MB236. I don't remember the complete. Another MB236 or yeah.
Okay. So that is the first outcome of this interdisciplinary conference now came out, because you are having that anacardic acid.
Yeah.
He was wondering about the cirrhosis
synthesize,
treatment, and it is also effective in cancer.
Yeah.
So that molecule is wonderful.
Yeah, we can give some samples and we can further detailed studies can be done.
Thank you, sir.
And if something comes out really useful, then we can go for a patent or commercialization.
Sir, your presentation was wonderful, and I think so many teachers are here who are doing research in pharmacognosy. We have a pharmacognosy wing actually, and anacardic acid is isolated from this shell of this cashew nut, and can we synthesize? Is there any possibility of synthesizing this anacardic acid? Because
but that has not been successful so far, because you can put a alkyl chain, but nature has created it with maybe 38 to 48% mono, one double bond, and around 20% two double bonds, and then remaining three double bonds like this. So that structural combination
it is
has
not possible. That is the wonderful work of the nature. Okay, that's isocitric acid was useful for the treatment of Kodambuli. Kodambuli in isocitric acid was extract ചെയ്തുകൊണ്ടിരുന്നു Sami chemicals. But recently isocitric acid was synthesized commercially. Is there any chance of synthesizing? Because it is a complex.
Citric acid and all is a relatively simple
molecule. This is a complex molecule like vincristine or vinblastine, which we were not able to synthesize. So anacardic acid, we can do further research in anacardic acid and isolation techniques. Simple techniques can be formulated. And you isolated anacardic acid, right?
We have
from
isolated and characterized.
And you developed that system for isolating it, right?
Yes.
Is it published in literature?
No, that is.
Yeah, you have to publish it, and then only you have to.
Yeah, we will do it.
Okay,
soon.
sir. It's a very excellent work, and an example of this interdisciplinary research he has done on HeLa cell lines, that is cervical cancer cell lines. It is effective. So we can do further research, and collaborative research can be done with pharmacognosy or pharmacology people can help you out in this area. Thank you so much, sir. Wonderful, your wonderful question.
Thank you, sir. Now we would like to call Dr. K. Suresh to present certificate to Dr. Dinesh Kumar. We would like to call Dr. Agash Maradagam, Professor and HOD, Department of Pharmaceutical Chemistry, National College of Pharmacy, to present certificate to Dr. Dinesh Kumar.
Now, let us start the second session on the topic Advancing Pharmacy Education and Research, a visionary perspective on interdisciplinary approaches at Kerala University of Health Sciences by Dr. Rajashree R.S. I would like to invite Jyoti M. Joy, Professor and HOD of Pharmacognosy, National College of Pharmacy, as the chairperson of the session. Jyoti ma'am has almost 15 years of teaching experience, and she has published various research articles in many national and international journals. We welcome you, ma'am.
Good morning, everyone. A warm welcome to the prestigious session on advancing pharmacy education and research, a visionary perspective in interdisciplinary approaches at Kerala University of Health Sciences. It is with great pleasure and honor that I stand before you today, and I have the privilege of chairing the session. I would like to introduce our distinguished speaker for today's session, Professor Dr. Rajashree RS. Dr. Rajashree holds an impressive academic background with a bachelor's degree and master's degree in pharmacy from University of Kerala, Thiruvananthapuram, and a PhD in pharmacy from MG University, Kottayam, Kerala. With over 25 years of experience in the academic field, she has worked as lecturer, assistant professor, and associate professor in various government medical college pharmacy colleges. Currently, Dr. Rajashree serves as a professor in the Department of Pharmaceutical Chemistry at the College of Pharmaceutical Sciences, Government Medical College, Alapi. In addition, she holds the esteemed position of Dean of the Faculty of Pharmaceutical Sciences and Chairman of the Board of Studies of Pharmacy at Kerala University of Health Sciences, Thrissur. Dr. Rajashree's expertise extends beyond academia. She has obtained FE-GEC certification from the National Association of Board of Pharmacy, North America, and has undergone numerous training programs and seminars. She has also served as a resource person for several conferences and seminars, demonstrating her dedication to knowledge dissemination. With a strong research background, Dr. Rajashree has contributed significantly to the field. She had published over 20 research papers in reputed journals and has undertaken 2 research projects funded by SBMR. Her membership in professional associations such as the Kerala Pharmacy Graduate Association, Association of Pharmaceutical Teachers of India, Indian Society of Toxicology, and the Pharmaceutical Society of Kerala highlights her active involvement in the pharmacy community. Dr. Rajashree's contribution extends beyond her research and academic roles. She also serves as a scrutiny committee member of KUHAS for the approval of academic institutions, as well as member of the Senate and Academic Council of the University. We privileged to have you, ma'am, with us today, and I kindly request her to share her expertise and visionary perspective on advancing pharmacy education and research through interdisciplinary approaches at Kerala University of Health Sciences. Thank you once again, and let us proceed with the session. Ma'am, please.
Thank you so much for that nice words of introduction. See, I feel embarrassed when so much things are with words of appreciation comes. എന്നെക്കുറിച്ച് തന്നെയാണോ ഈ പറയുന്നതെന്ന് എനിക്ക് പലപ്പോഴും സംശയം തോന്നാറുണ്ട്. അപ്പം അത്രയ്ക്കൊന്നും വേണ്ടാന്ന് ഞാൻ എപ്പോഴും പറയാറുണ്ട്. ഓക്കെ, അപ്പോൾ നിങ്ങൾ ടുഡേ വി വിൽ ഹാവ് എ ഡിസ്കഷൻ. എല്ലാം നിങ്ങൾക്ക് എല്ലാവർക്കും അറിയാവുന്ന കാര്യങ്ങൾ തന്നെയാണ്. നത്തിങ് ന്യൂ വാട്ട് വി ക്യാൻ ഡു ആൻഡ് വാട്ട് വി ഹാവ് ഡൺ, അതിനെക്കുറിച്ചിട്ടാണ് നമ്മൾ. ആകാശ സ്ലൈഡ് ചേഞ്ച് ചെയ്യുന്നത് എങ്ങനെയാണ്? Page down. Okay. Alone we can do so little. Together we can do so much. Helen Keller. Helen Keller. She is a great inspiration to all of us. Helen Keller. Because she was blind, deaf and dumb, and she has done so many things. She developed Braille language for blind. So many things she has done. And what is research? And we are doing research every day. What is research? And definition of research is a systematic investigation, searching and finding new things, aim for developing contribution to generalizable knowledge. And it encompasses various fields including biomedical research, epidemiological research, health sciences research, studies of behavioral, social and economic factors impacting health. And what is the value of health research? We are doing health research, right? Anyway, health research means we are developing molecules. When we do pharmacognostic studies, we are isolating molecules. That's which the previous session, Dr. has given us a detailed description about a molecule which was isolated from this cashew shell, and that is also a research, and we can use that molecule for further studies. Disease strength, risk factors, treatment outcome, functional abilities, patterns of care, and healthcare costs, which we do. Pharmacy practice PGs usually do research on this area, and also clinical research approaches for complementary insights. Clinical trial, all the drugs which we are releasing into the market has to be go through the various steps. You all know the steps which we do, and we have to go to clinical trial, preclinical studies, and then to clinical trials, and we can help, and we can do research on clinical trial on efficiency and adverse reactions of medical interventions, and real world clinical experience aids in comparing and improving drug, vaccine device, and diagnostic use. In developing drug, it takes several years for releasing a drug or vaccine into the market. But recently, without any research, without much knowledge regarding the safety, we have released a vaccine into the market, right? COVID vaccine was released because of that demand of this society. The COVID vaccine was released without much clinical research, and the duration was waived actually, and the drug was released into the market. The vaccine was released into the market. And what is the importance of clinical experience? And this FDA approval, to get a drug approved, we should have a approval from FDA on controlled clinical trial, but real world data is essential for clinical use. Preclinical studies, we do preclinical studies, and clinical studies are done in a small group of volunteers, and then after it is released into the market, and when it is released into the market, that for clinical use, real world data is necessary. And cracking clinical experience helps to identify rare adverse effects and determine effectiveness in different populations or circumstances. So many drugs were released into the market, and it was withdrawn from the market. Nemucilide was recently released. Roxicosib was withdrawn from the market due to the cardiotoxicity. So similarly, after releasing into market also, due to the clinical trials or due to the adverse effect that drug produce, it is withdrawn from the market, and supports the development of guidelines for best practices and high quality patient care. And there is a trend of misuse of antibiotics. 1 or 2 days antibiotics is used, and then it is abruptly stopped by individuals, and that resulted in development of drug resistant varieties of bacteria. That is the main problem. Multiple drug resistant TB is it's a huge burden to our society as a whole. Multiple drug resistant TB, it cannot be treated with any drug available in the market, and they are isolated. Actually, even now they are isolated and treated since, but this actually this treatment is not available, and this is due to irrational use of these antibiotics or drugs which were used for the treatment of TB. And what is the impact of health research? Significant development and discoveries of new therapies. New therapies can be introduced. Remarkable improvement in healthcare and public health can be done by research. Only by research we can do that. Contributes to human health, longevity, and increased productivity, benefiting the national economy. The life expectancy has increased a lot. Why this life expectancy has increased from 60 to 80, 85? It has increased only because of the medical research, and the patients and doctors are more concentrating on the health of the individual, and all these are because of the contribution by the medical field. And the impeding or weakening the research enterprise negatively affect the social interest, and if the research fails, then the social interest will also fail. And what is collaborative research? We are focusing on collaborative research now, means working together and sharing ideas between different areas of healthcare. It helps pharmacy education by teaching students about how healthcare work and how to care for patients in a team, and how as a collaborative effort we can do research. By collaborating in research across different fields like chemistry and medicine, we can create safer and better medicines. We can work together in developing new drugs, understanding how drugs works, and making sure students learn about medicine in a well-rounded way. Actually, we are doing research as a pharmacy postgraduates, and even in undergraduate level we are doing research and new biomolecules. We are selecting a particular plant. When we do a research on phytochemical research, we are selecting a particular plant and we are isolating a drug from that plant. But that even that area, we need a scientific evidence that this plant is effective in these diseases, and we usually depend upon Ayurveda. Actually, Ayurveda, this plant is used in a preparation in Ayurvedic preparation. This plant is one of the ingredient in Ayurvedic preparation, which is meant for anti-inflammatory activity. So, but in the main difficulty, the Ayurveda was developed thousands of years back, and the modern medicine is not having that much history. And Ayurveda, in Ayurveda, it is mentioned in the books of Ayurveda, and we have to prove that. We have to have an idea whether it is effective in that disease, and we will try to isolate a particular compound, and we are trying scientifically, we are proving that why they use that particular ingredient or particular plant in that Ayurvedic medicine. And most cases when we do research on a particular plant which is used in Ayurveda, we are getting promising result, and we should have a scientific basis for selecting a particular plant for doing the research, and that is being contributed by Ayurveda, Unani, Siddha, and ancient Chinese system of medicine, and we can follow that. And only from that we are getting the idea of how which part of the plant or which species of the plant is used, and that can be beneficial to do the particular research. So we can work together to developing in new drugs, understanding how drug works, and making sure students learn about medicine in a well-rounded way. And the collaboration with healthcare streams, improve patient care, leads to new discovery, helps students become better healthcare professionals. And what areas we can do collaboration with Ayurveda College, formulation development. Pharmacy College can assist in formulating and standardizing Ayurveda products, ensure quality, safety, and efficacy standards are met. The medicine preparation of Ayurveda is very complex. Even they will pay attention to the vessel in which they make the Ayurveda medicine is important. The wood they use for preparation is also specified in the literature, because when we prepare a particular medicine in a particular vessel, it is having a specific heat of each material is different, and it will provide a particular temperature. Above that temperature, if we use another vessel, the temperature will go above that, and that will cause deterioration of the active ingredient. Even we cannot find in the Ayurvedic literature, even though it is not given, why the reason is not given, but only when we prepare the medicine as given in the literature, it is going to give the 100% result. But for each category, there is each procedure which if they follow in Ayurvedic medicine, there is a reason, and we should find out the reason why they follow that particular procedure. We should find a reason. And for standardizing Ayurvedic products, now we have HPTLC methods are there. We can identify a marker compound, and there is a marker compound which is available in a particular formulation, and using that marker compound, we can find out whether that marker compound is available, and we can assure that it is of standard quality. Even then, it is a very difficult task for standardizing Ayurveda products because of the complexity of the phytochemicals and phytomaterials which we use. It makes it a very complex phenomena or procedure. And then quality control and analysis. of Ayurvedic medicine can also be done, and consistency of preparation. When we manufacture an allopathic drug, or even if it is manufactured in National College or in Government TD Medical College and Alappuzha, it will not vary much. There is no considerable variation. There may be variation, 1 or 2% variation may be there. But when we prepare an Ayurvedic product, the consistency we cannot ensure. The consistency, and it may vary. A drastic variation can occur in case of Ayurvedic formulation. And so recently this Ayurveda system of medicine also has come up with new formulations, like Ayurveda capsules are available, Ayurveda tablets are available. New formulations are developed in case of Ayurvedic products, and that in formulation development, pharmacy graduates can very well help in developing new formulations, and also quality control and analysis, and which can ensure the safety of Ayurvedic products. Now we have Ayurveda B.Pharm also in Kerala University of Health Sciences. They can help in ensuring the safety of the Ayurvedic products. And drug discovery, I have already mentioned about knowledge combined with scientific method for new drug discovery, identification of active compounds. If a particular plant is used in a particular formulation in Ayurveda, then we can select that plant, and we can do for which disease that plant is used. We can do research, and we can identify the active compounds, and we can further develop therapeutic agents. That is in the CSIR, the research scientists have done from cashew, and he has developed that is used as a parent molecule, and derivatives can be prepared and it can be used. And in research projects can be done with Ayurvedic medicine and modern research methodologies, and phytochemical analysis, drug delivery system, and herb interactions, and to find out whether a drug herb interactions are going or are happening when these medicines are taken. Then pharmacokinetic and pharmacodynamic studies can be done for Ayurvedic medicine, and it can bridge the traditional Ayurvedic practices and scientific understanding. And we can also do clinical trials with Ayurveda and pharmaceutical science, collaborates on ethical and complement clinical trials, generate scientific evidence supporting efficiency and safety of Ayurvedic medicine, Ayurvedic evidence-based practice. In modern medicine, it is based on evidences, and even though we have experience from the traditional use of Ayurvedic medicine, in some cases for scientific evidence which we depend in this modern period, everything needs scientific evidence. That scientific evidence is not available in certain cases in Ayurveda, Ayurvedic system of medicine. And in developing scientific evidence and supporting the efficiency and safety, we can assist them, and we can collaborate them with doing that. And then formulation development. Pharmacy colleges, in Trivandrum Medical College, we usually, the PG students from Government Ayurveda College usually visit our college, and PG students and industry present to develop modern dosage form like capsules, tablets, creams, suppositories of their medicine is developed, and it acts as a collaborative work in our college. Then standardization, joint efforts in quality control and standardization of crude drugs and Ayurvedic medicine. Standardization can be done by collaborative research, education and training. Sciences offers specialized courses to integrate knowledge, creates professional well-versed in both traditional and modern medicine, and public health initiative joint efforts in developing evidence-based guidelines for Ayurvedic medical use, promotes rational use, educate healthcare professionals, and enhances healthcare option. And to summarize, Ayurvedic principle and pharmaceutical sciences can bring together traditional wisdom and modern scientific advancement through joint effort in formulation development, drug discovery, quality control, research, education of medicines. it is a truth where they are very reluctant to accept this other alternative system of medicine. But invariably, the alternative system of medicine is very effective and very simple in treating so many diseases. So many diseases are treated, skin diseases in particular. There are good options in pharmacy, this Ayurvedic system of medicine. They can cure skin diseases, and there are a few opportunities in case of modern medicine for treating the skin diseases. But we pharmaceutical, we the students and faculty and graduates of pharmaceutical sciences are welcoming other disciplines also, because we understand that the traditional system of medicine has much more than the new, much more to contribute than the new modern medicine. And Ayurveda, in Ayurveda, this thailas, external application for various diseases, arthritis, external application is done. And only 40 or 50 years back, that concept of percutaneous absorption was understood by the modern medicine. Percutaneously the medicine is being absorbed എന്നുള്ള ഒരു കാര്യം പോലും മോഡേൺ മെഡിസിൻ ഒരു 50 വർഷം മുമ്പാണ് കണ്ടുപിടിച്ചതെങ്കിൽ thousands of years back, that concept, that the medicine is absorbed percutaneously എന്നുള്ളത് ആയുർവേദിക് സിസ്റ്റം ഓഫ് മെഡിസിനിൽ അറിയാമായിരുന്നു. And all the modern medicine give scientific evidence, and that scientific evidence we have to help the pharmacy students can, or pharmacy researchers in pharmaceutical sciences can help to understand and to give evidence that this is what is happening and why this drug is producing effect. This is the reason why this producing effect. And in homeopathy is a totally different concept. When the dilution increases, the potency increases in case of homeopathic medicine. And when if we take and test homeopathic medicine is very having 100 potent or 1000 potency, it will not have any drug molecule available in the when we do a chemical test. And there are homeopathic tinctures also. But when we take the homeopathic tinctures, we can do we can analyze what are the ingredients present in homeopathic tinctures, what are, whether it contains phenolic compounds, whether it contains these flavonoids or alkaloids, glycoside. That part we can analyze, and we can find out whether it is due to this particular compound this homeopathic medicine is producing result, so that we can validate with scientific evidence that this drug is homeopathic system is true, homeopathic system is effective because of the presence of this particular compound. And that in that area we can do research also, and we can take the help of homeopathic doctors regarding the treatment options they have for a particular condition. The tinctures they use for this antihypertensive or hepatoprotective or renal protective activity, and we can scientifically prove that these compounds are having activity. So joint research to understand the mechanism of action of homeopathic medicine, pharmacological effect, therapeutic benefit, safety and quality assurance area we can do collaborative research with homeopathic discipline, research on potency and dilution, of potency and dilution in commerce, this homeopathic medicine. We can do education and training, join workshop or training programs to enhance knowledge, until we can have knowledge regarding the homeopathic system of medicine, homeopathic from pharmaceutical sciences, and pharmaceutical science students can gain insights into homeopathic principle and practice, and collaboration in homeopathy and pharmaceutical sciences facilitate research on potency and dilution, insights of highly diluted preparations, and enhance education and training opportunity for students in both fields. We are talking about collaborative research. എല്ലാവരും അങ്ങ് ഉറക്കം തൂങ്ങിയിരിക്കുന്ന പോലെ എനിക്ക് തോന്നുന്നു. സോ മെഡിക്കൽ കോളേജസ് കൊളാബറേറ്റീവ് റിസർച്ച് മെഡിക്കൽ കോളേജസ് ആകുമ്പോൾ കുറച്ചും കൂടെയൊക്കെ നമുക്കറിയാം മെഡിക്കേഷൻ മാനേജ്മെൻറ്റ് ആൻഡ് ഓപ്റ്റിമൈസേഷൻ we are a part of this medical college or medical fraternity, we can say. Role of clinical research in hospital, expertise in drug selection, dosing and monitoring, collaboration with medical college for safer and effective medication management. Actually, pharmacists are not accepted എന്നുള്ള ഒരു ജനറൽ കോൺസെപ്റ്റ് ഉണ്ട്. ദാറ്റ് ഈസ് ട്രൂ ഫോർ എ സർട്ടൺ എക്സ്റ്റെൻറ്റ് ഓൾസോ, ബട്ട് വി ഹാവ് ടു പ്രൂവ് ദാറ്റ് വി ആർ qualified, we have to prove that we are efficient, we have to prove that we are experts of medicine. എങ്ങനെ പ്രൂവ് ചെയ്യുന്നേ? വി ഹാവ് ടു ഗ്യാദർ നോളേജ് ആസ് മച്ച് ആസ് വി ക്യാൻ. നമ്മുടെ നോളേജ് ഉണ്ടെങ്കിൽ മാത്രമേ നമ്മളെ ആരും വില വയ്ക്കുകയുള്ളൂ. അല്ലാതെ വെറുതെ ചെന്നിട്ട് നമ്മൾ ഫാം ഡി ആണ് അല്ലെങ്കിൽ എം ഫാം ആണ് അല്ലെങ്കിൽ ബി ഫാം ആണെന്ന് പറഞ്ഞ് നിന്നാലേ ദേ വിൽ നോട്ട് അക്സെപ്റ്റ് അസ്. വി ഹാവ് ടു ഇൻ്റർവീൻ, വി ഹാവ് ടു, ഒരാളിരുന്ന് നല്ല ഉറക്കമാണേ, വി ഹാവ് ടു, ആരാന്ന് പറയില്ല, we have to intervene, we have to intervene, and we have to go and we have to contribute. ഒരു പ്രശ്നം ഉണ്ടാകുമ്പോൾ, when there is a problem, we have to contribute our knowledge, and then only we get acceptance in the society, and then only we will get an acceptance among the medical fraternity. So medical review and adverse event monitoring, മെഡിക്കേഷൻ റിവ്യൂസ് ഫോർ പേഷ്യൻസിന് വേണ്ടി ഇത് ചെയ്യാം, ഇതെല്ലാം നമ്മൾ പഠിക്കുന്ന അല്ലേ. വെൻ വി സ്റ്റഡി ഫാർമസി പ്രാക്ടീസ്, ഐഡൻറ്റിഫൈങ് പൊട്ടൻറ്റ് ഡ്രഗ് റിലേറ്റഡ് പ്രോബ്ലംസ്, കോൺട്രിബ്യൂട്ടിംഗ് ടു ഫാർമക്കോ വിജിലൻസ് ആൻഡ് എൻഹാൻസ് പേഷ്യൻ്റ് സേഫ്റ്റി. So when the pharmacy practice PG students go for ward round, they should, previous day itself they should learn what are the patients who are admitted in the hospital, what are the possible treatment drugs which are given for the treatment, what drugs they have given, and is there a possible interaction, is there any alternative drug available. ഇതിൻ്റെ കാര്യമെല്ലാം we have to learn, we have to study, and we have to go there after having a thorough study and thorough knowledge about that particular disease and particular patient and patient's condition. Then only we can contribute. So you try to enhance your knowledge as much as you can, and then only we will get an acceptance. And അത് ആരും ഒന്നും നമുക്ക് ഫ്രീ ആയിട്ട് തരില്ല. We have to fight for it, and we have to work for it. Okay, conducting medication review for the patient, identifying potential drug related problems, contributing to pharmacovigilance and enhancing patient safety. ഇതൊക്കെ ചെയ്യാൻ പറ്റും, provided we have knowledge. We have knowledge in that particular area. So try to increase your knowledge as much as you can. Okay, and therapeutic drug monitoring. What is therapeutic drug monitoring? What is monitoring? What is monitoring? ഇതെല്ലാം നമ്മൾ പഠിക്കുന്നതാണ്. Closely watch a drug for its side effects, for its overdosage. Overdosage produces toxicity, and what is there are lab reports. We can verify the lab reports. All those things can be done, and then we can do. And ensuring optimal drug dosage through monitoring. We can increase the dose or decrease the dose where any renal problem is there, any this hepatic problem is there, whether they are taking any drugs concomitantly. All these aspects we can learn, and personalizing treatment plans based on individual patient needs. That is the ultimate aim of therapeutic drug monitoring. Advancing clinical pharmacy practice, studies on medicine, ഇതെല്ലാം തന്നെയാണ് ഞാൻ നേരത്തെ പറഞ്ഞത്. Studies on medication effectiveness, safety and outcomes, generating evidence for evidence-based prescription practices, and improving patient care. Collaboration between clinical pharmacists and pharmaceutical scientists, providing update to data, evidence-based information on medications, tailoring drug information for healthcare professionals and patients. Patientsine മരുന്ന് എങ്ങനെയാണ് എടുക്കുന്നത്. Even now the medicines are not taken, especially for pediatric patients. One of your one of the faculty is doing research on the pediatric patients. She is planning to do research in pediatric patients, and these pediatric patients, their compliance is very less, and they are taking wrong medicines. Invariably they are taking wrong medicines, and it's an area where we can contribute, and also we can also help providing training and continuous education, we can also contribute and collaborate with them. And for specialized courses on pharmacotherapy, clinical research, and pharmacoeconomics, we can take. And now we are planning to, in our university also, we are planning to start certificate courses for the students, so that it is easier for the to pursue for job easily after getting this certificate. And clinical pharmacy updated and contribute to evidence-based medicine. And clinical assisting, assisting medical professionals in therapeutic drug monitoring, clinical toxicology, pharmaceutical, pharmacovigilance, etc. We have PharmD graduates and PharmD students. They are doing their clerkship, and they are doing their ward round, and they can help medical fraternity regarding this safe pharmacovigilance and toxicology. And drug discovery and development, also they can collaborate between medical and pharmaceutical sciences. Medicinal chemistry, pharmacology, formulation development, all is integrated. Actually, when we do research in pharmacy medical college, anatomy is doing research in anatomy, biochemistry is separate, medicine separate, cardiology separate, but in pharmacy we cannot do it. Is very difficult to do in a particular area. When we develop a new drug molecule, we have to do the effectiveness, we have to do certain in vitro cell line studies, and we will find out that this drug is effective. Then we will go for animal studies, and that animal studies involve pharmacological studies, animal studies, and then only we can confirm that up to preclinical studies this drug was found to be effective. And translating scientific discoveries into new medications and therapies. And interprofessional education, enhanced medication management, advances in drug discovery and development. Then comes the collaboration with dental college. Here in National College, they are doing, your principal was telling me that they are doing collaboration with the dental college in research and formulation development, that is oral solutions, specialized formulation for dental drugs, etc. Certain dilutions. When I was a student in government medical college, dental PG students used to come to our college to get their drug diluted for dental use. And in that area we can help them. Oral films, patches, development of oral films, dental patches, gels, and innovative drug delivery systems can be developed by collaborating with dental faculty, and also enhance the effectiveness of patient compliance and convenience of dental treatments. Local anesthetics, there's lignocaine and the adrenaline, that is a classical drug which is used for the nerve block anesthesia, and novel delivery system for local anesthesia can also, we can develop novel drug delivery systems, controlled release systems, and innovative techniques for this anesthesia, local anesthesia. Different formulations can be developed. Efficacy and duration of local anesthesia in dental procedure, it's possible to develop new formulation, and we can help out in that area. And also pharmacokinetic studies, collaborative research on pharmacokinetic dental drugs, dental asbestos combined with analysis of drug absorption, whether this drug is absorbed into the human body. That in vivo studies, that is animal studies, also can be done. Drug absorption, distribution, metabolism, and excretion of drugs which is used for dental purposes can be studied using by collaborative research with dental and pharmacy college, and also optimize drug dosing and treatment regimes in dental medicine. Antibiotic resistance in dental practice. My colleague Dr. Dilip was discussing yesterday with me that most widely used antibiotics are most widely used for dental abscess, for extraction procedures, everything. These antibiotics are very, use of antibiotics is high, and the prescription of these antibiotics, whether it is rational or not rational, no such studies. Even in medical colleges we do the studies, we do this patient prescription analysis, everything is done in dental medical colleges. But in dental colleges, we are not doing that, whether it is abruptly being stopped. And that type of studies can also be done by these pharmacy practice students when they do PG postgraduate program. They can focus on this dental sciences also, whether there is an incidence of abruptly stopping this antibiotic, or whether any effect is produced by stopping when a dental procedure is being carried out. This anticoagulants which a cardiac patients take is abruptly stopped, and then they do the procedure. That type of study, you can find better options, and that kind of studies can be done. And exploring alternate antibiotic strategies in dentistry can be done. Pharmacotherapy education in dental curricula, collaborate in education dental programs. We can collaborate with them, and we can give ideas regarding the pharmacotherapy, prescribing medication, managing drug interaction, and recognizing adverse effects. All this area we can help out, and we can also go hand in hand with them for a better compliance and better efficiency in patients. Interprofessional collaboration between dentists and pharmacists will be very useful for the society as a whole. And dental materials research. Dental materials, National College has already started research on dental materials, better material for this cavity filling, better materials where there is any dental adhesive, enhancing the properties and performance of dental adhesive, restorative materials, biomaterials, composite filling in ഉപയോഗിക്കുന്ന materials. All these can be, newer material development can be done on collaborating with the dental college and incorporating antibiotic agents or growth factors. Whether it can be incorporated along with the composite filling material, that all type of studies can be done without losing the efficacy. MLT and other paramedical streams. Okay, medical laboratory collaborative design and optic diagnostic assay techniques. We are developing these new analytical techniques for drugs and pharmaceuticals, and we can also try out analysis. The PG students can try new development of new assay for certain for testing certain things, that is urine analysis or blood analysis. Different techniques can be developed by using simple techniques available, color test rather than other simple techniques can be developed, and to improve the accuracy and efficiency of diagnosis. Diagnostic procedure can be improved, and drug interactions and toxicology studies can be performed with the help of medical laboratory technician and experienced in an joint research on drug interaction and toxicologically can be done. Experimental analysis and pharmacokinetic and toxicology expertise can be used, and biomarkers can be developed for particular diseases, and it can be validated. Novel biomarkers for disease detection. Biomarkers usually maybe detect this disease or diagnose a disease using biomarker. Development of new biomarkers is a possibility for this pharmaceutical related. So a sample connection and analysis with pharma is done, and we have of course we have to get ethical. There are so many issues, ethical committee clearance, all this we have to get, and after that we can do biomarker research. Okay. Quality assurance and quality control, standardized procedure for accurate testing. So there is a standard protocol for laboratory testing, and we can ensure that there is standardized protocols for accurate laboratory testing are done, and implement quality control measures for reliable research. And emerging technologies in diagnostics, that is what I was mentioning, integrate innovative technologies into laboratory practice, MLT practical implementation with the pharmacy scientific expertise. We can develop, and that using our that we can collaborate with their practical experience, and we can develop and design novel diagnostic procedures for diagnosis of certain disease. And then collaborating with nursing stream, medication safety, usually research into minimize medication errors, because in our pharmacy, in our hospitals, nurses are dispensing medicines, not pharmacists. I have observed that, because most of the cases the drugs are given by the nurses, not by the pharmacist. For the patients, drugs are given by the nurses. And this research to minimize medication error, we can help them to educate, and we can help them to develop protocol for safe medication administration. And then joint programs can be conducted to promote collaboration between nurses and pharmacists, and enhancing patient care through interdisciplinary teamwork. And also medication therapy management process, we can work hand in hand with the nursing professional for optimizing medicine regime and patient outcomes. Drug effectiveness, evaluating the efficiency and safety of medication. This is similar, almost similar to what we do with the doctors, and also we can recommend for optimal medical use. And we cannot direct them, okay? We cannot direct them. നമ്മൾ അങ്ങോട്ട് ഡയറക്റ്റ് ചെയ്യാൻ ചെന്നാൽ അവർ സമ്മതിച്ചും തരില്ല. So we can give recommendation for optimal medical use. And also collaborative effort to educate the community on medication management, conducting health screenings and promoting health awareness, and development of clinical guidelines and protocols. The development of protocols, we can help out, and creating evidence-based guidelines for medication management and enhancement of person safety, and standardizing care practices. And veterinary streams. Veterinary medicines are also used like human medicines. Development of veterinary pharmaceuticals, we can help. Comparative pharmacology and pharmacotherapeutics, pharmacokinetic and drug absorption studies in animals can be done. Antibiotic resistance and antimicrobial stewardship, we can do. All this we can watch, or we can monitor this antimicrobial studies, whether antibiotic resistance is equally applicable to human beings and veterinary practice. Veterinary drug safety and adverse effect monitoring, we can do, we can help, and we can do collaborative project with the veterinary sciences. And the concept of one health approach with your Nawasar, your managing trustee, has in his presidential address has given an insight about this one health approach. It is the newly emerging system, newly emerging concept, I should say, that is human beings, animals, plants, all are considered together as a single unit, and treatment is given in that approach. And collaborative research impact, discuss on potential impact on animal health care and public health, highlight benefits such as evidence-based veterinary medicine, improved treatment outcomes, and preservation of antibiotic efficiency. Antibiotic resistance and antibiotic stewardship, adversing global change of antibiotic resistance, collaborating, investigates antibiotic stewardship in veterinary medicine. We should monitor and closely observe. These antibiotics are given in a similar way to this animals also. So many diseases, there's cattle, these domestic animals, dog, cat, and they are treated as a member of the family nowadays, right? Cats and dogs are treated as a member of our family, and they are treated in a better way than human beings now. So they are given all the vaccination procedures. They are following all vaccination procedures. They are given. They are these cats and dogs, when they get a skin infection, they are being treated with antibiotics. So all the treatment options applicable. Nowadays, surgeries, complicated surgeries are being conducted in this veterinary hospitals, and veterinary private hospitals has emerged. So effectiveness of existing antibiotics in these animals is equally important as human being, and adverse effect monitoring and veterinary drug safety is important, and we can collaborate them, and we can do research. Interdisciplinary research can be done, and one health approach, embracing one health approach, the collaboration recognizes the interconnections of human, animal, and environmental health, and researchers investigate zoonotic disease, drug-resistant pathogens, and impact of veterinary pharmaceuticals on environment. Because these cattle, when they consume, they are excreting that cattle cow dung is used for as a manure, and whether it is being absorbed by the plants, and whether it is coming in the grains which we consume, which we do not know. Okay, so that type of research also can be done by collaborating with veterinary pharmaceuticals, and impact of this veterinary pharmaceutical on environment can also be studied. And in my session, I have tried to give a insight regarding importance of collaboration, fostering a holistic approach, benefits of interdisciplinary collaboration, advancement in pharmaceutical research and education, and paving the way for a comprehensive healthcare system, vision for advancing pharmaceutical education and research. So recently, Jharkhand government, Chief Minister of Jharkhand was giving a speech, and they have lifted the need of pharmacists for dispensing a medicine. So the concept of pharmacist and medicine, which we used to have right from 1947, is gradually changing, and we have to change our role of simply dispensing a medicine, and you never be satisfied with 15,000 or 20,000 rupees which you get from the hospital, and you have to decide what you have to do, how we can improve, and how we can get a position in the society that we are also indispensable as a member of the healthcare team. For that, you students should work hard. Everybody can do that. Everybody can contribute for making this world a better place to live in. I thank once more for inviting me to give a session. Always they will invite me for inauguration. To give a keynote address, then inaugural speech, and this time I told Agash, when he invited me, I will conduct a session. I like to conduct a session because teachers always like to do that rather than inaugurating like politicians. So it was a very wonderful experience in interacting with you. If you can share any idea with me, you are welcome. Thank you so much.
Thank you, Rajashree ma'am, for your informative session. Now it's time for doubt clearing.
You can share your knowledge. Nothing new information was not given. You can share your idea. Okay.
And thank you, ma'am, for your informative session. And madam has explained us about the importance of scientific evidence and the research, and the possibilities of collaborative research, and how it helps our healthcare system, and what, as a pharmacist, what we can do in the research and other things. So thank you, ma'am. And now it's a time for the delegates to ask if you have any queries or questions or anything. So please ask, ma'am, and you can clarify your doubts.
In the homeopathy, madam told that is dilution increases the potency.
Yeah.
Can we compare it with the nanoparticles or?
Yeah, when this homeopathic system is totally different. Actually, in modern medicine, the treatment is we treat a particular disease or particular organism or particular condition. Treatment is given. But in Ayurveda also, the system is taken as a whole. അല്ലേ അങ്ങനെയല്ലേ ഒരു വ്യക്തിയാണ് അവർ ട്രീറ്റ് ചെയ്യുന്നത്. ഇപ്പോൾ എനിക്കൊരു അസുഖം വന്നു, ആ അസുഖത്തിന് തരുന്ന ചികിത്സയല്ല വേറൊരു ആൾക്ക് സെയിം അസുഖം വരുമ്പോൾ. അപ്പോൾ ആ ഒരു അപ്രോച്ചാണ് ഹോമിയോപ്പതിക് മെഡിസിനും. ഇപ്പോൾ ഒരു അസുഖമായിട്ട് ചെല്ലുമ്പോൾ ആ അസുഖത്തിന് തന്നെ അല്ല ട്രീറ്റ്മെൻ്റ് അല്ല. അതിന് ഒരുപാട് തിയറീസ് ഉണ്ട്. നമ്മുടെ ഈ ഒരു മോളിക്യൂൾ ചെന്നിട്ട് ഡൈല്യൂട്ട് ചെയ്യുമ്പോൾ അതിൻ്റെ ഒരു ഇംപ്രഷൻ ഉണ്ടല്ലോ, മോളിക്യൂളിൻ്റെ ഒരു ഇംപ്രഷൻ ആ സോൾവെൻറ്റിൽ വരും എന്നാണ് പറയുന്നത്. ആൻഡ് വെൻ ഇറ്റ് ഈസ് ഡൈല്യൂട്ടഡ് ദ impression does increase, that memory of that molecule remains in the solution, and that when it is diluted, that is increasing, it is multiplying. Actually that impression in the solvent will go and bind with the receptors and it produces the action. അങ്ങനെയുള്ള ഒരു തിയറി ഉണ്ട്, but there is no scientific evidence to prove that. That is the thing. അതാണ്, it is very complex. That is the difference between this അലോപ്പതിക് സിസ്റ്റം ഓഫ് മെഡിസിൻ ആണെങ്കിൽ നമുക്ക് പറയാൻ പറ്റും, ഇങ്ങനെ ഇതുമായിട്ട് കമ്പൈൻ ചെയ്യുമ്പോൾ ഇതിൻ്റെ ഈ എൻസൈമിനെ ഇൻഹിബിറ്റ് ചെയ്യും, അല്ലെങ്കിൽ ഈ എൻസൈമിൻ്റെ പ്രൊഡക്ഷൻ കൂട്ടും, പിന്നെ പാർക്കിൻസോണിസിൻ്റെ ഡിസീസിൽ ഡോപ്പായുടെ കോൺസെൻട്രേഷൻ കൂട്ടും, അങ്ങനെ ബ്ലഡ് ബ്രെയിൻ ബാരിയർ ക്രോസ് ചെയ്യും എന്നൊക്കെ പറഞ്ഞിട്ട് നമുക്ക് കാര്യങ്ങൾ റീസൺസ് പറയാം. പക്ഷേ ആയുർവേദിക് സിസ്റ്റത്തിലോ ഹോമിയോപ്പതിക് സിസ്റ്റത്തിലോ പറയാൻ പറ്റില്ല. പക്ഷേ ഇറ്റ് ഈസ് എഫക്റ്റീവ്. അത് the physicians know and the patients who experience know, but there is no scientific evidence. That is the thing.
One of the thing about this homeopathy is again that in that the genius of homeopathic medication, one of the textbooks they are following, it says that when 1 lakh enunciation, that is a dilution, 1 lakh enunciations goes on, the number of molecules, active molecules in that preparation will go below the Avogadro number, so that we cannot find out that molecule. But for dilution, they are using two things. One is lactose and second is alcohol. And they are considering these are neutral molecules. And when these neutral molecules are interacting with active molecules, some sort of charge is transferred to the neutral ones. And as the active molecule reduces, the number of molecules, the inactive molecules increases. The charged molecules increases. And that is also some based on the physics. They are not based on the chemistry and receptor interaction. They are based on some physics. But that theory is not yet proved. They says that it is only a black box effect. Something is given in, something is coming out. But what is happening in that box, nobody knows. Thank you.
Any more queries? Okay. I would like to express my gratitude to Dr. Rajashree ma'am for sharing her valuable insights, and to all of you for your active participation and engagement. Thank you.
Now we invite Jyoti ma'am to present the certificate for Rajashree ma'am. We invite Dr. Shamnas, Principal, KMCT College of Pharmacy, Kuttipuram, to present the certificate for Jyoti M. Joy.
Attention. Now we can break for our lunch. next session will start at 2 p.m. and the lunch is arranged at Pharmacy College. Those who want to avail the bus service, the Pharmacy College bus will be available from the Dental College campus at 12:30. So the lunch break will start at 12:30 to 2 p.m. The next session will start exactly at 2 p.m. Thank you.
Habiba KK, Professor KMCT Ayurveda Medical College, as the chairperson of the session. She is having more than 10-year experience in academic. She is the member of World Ayurveda Foundation and member of Vignan Bharati. She has published various scientific papers in national and international journals. We invite you, ma'am, for chairing the session.
Good afternoon, all. It's very nice to see you all today. Today is such a wonderful day, Guru Purnima day. So having a scientific session on this day is great. And thank you, Oma sir, for choosing me for this. So we have a wonderful speaker here. Before that, I don't know who has selected this topic, maybe sir only, right? But yeah, both the things are there, a traditional knowledge system is to be validated as a question, isn't it? That itself is a question. But that's true, because you go to China and talk in Malayalam, nobody will understand, doesn't it? So we speak in terms of Vata, Pitta, Kapha, and we say nobody is listening to us. It's true, we should talk in a way that others should understand. So the role of people like you is most important in this era, because people find that chemicals and synthetic things cannot meet all the requirements, health requirements today, and people are turning slowly towards natural products. But we have a treasure with us, but we are unable to tell how it is acting and what is the mode of action, what is the way it is entering into the body. And I cannot say 100, maybe thousands of chemicals together in a combination entering to the body and working miraculously. That's why people say Ayurveda is magic. But it's not magic, it's logic. Magic never repeats, logic repeats. Ayurveda repeats, so it is logic. But we cannot explain what is the logic behind it to the common public or the modern era people. So I think we need to have a very thorough knowledge of how you are validating it. Even I am very much interested in the newer techniques, how you are doing it. And to say about sir, I had, went through his CV and I had lost my consciousness, because there is a lot to speak about him. So few points I'll tell you for your understanding, because you should understand who is speaking all these, otherwise you will not get to know its gravity, isn't it? So Dr. C.T. Suleiman sir, he is senior scientist of phytochemistry division in CMPR, Kottakkal Ayurveda Shala. And the most important thing is his areas of research, and he is concentrated in phytochemical characterization of medicinal plants and secondary metabolites, and scientific validation of traditional knowledge and ingredient analysis. His research goes ahead to isolation of many phytochemicals, including 6 new molecules and 35 new reports from various medicinal plants. And now he focuses on herbal antiviral drug for SARS in collaboration with CSIR, and he also handles many collaborative works. And he has more than 60 research articles to his credit, and he has guided more than 50 PG theses, and he is board member of Kannur University and reviewer of many renowned journals. And he has also received Swami Vivekananda Yuva Pratibha Award and Young Scientist Award by Government of Kerala, and Dr. P.D. Sethi National Award for Best Research Article in 2020. So it's my great privilege to welcome you, sir, to the dais. Welcome, sir.
Good afternoon to one and all, and thank you for the nice comments of our respected chairperson. And first of all, let me to extend my sincere gratitude for inviting me this program, particularly your respected principal Verma sir. and the coordinator of this program. So, as in the introductory remarks, our chairperson indicated, this is a topic related to the validation of traditional knowledge using the modern scientific techniques. So, my presentation might be related to the Ayurveda, definitely as Madam indicated that the modern scientific terms are required to establish the scientific integrity of this traditional knowledge, because all these traditional knowledge bases are written in thousands of years back. Still it is in practice and it has the evidence-based clinical practice for many disease conditions, even in the case of the pandemic era also. So this is the outline of my presentation, starting from the traditional knowledge base, and of course the standardization is required if it is any practice or any systems of medicine. Then scientific validation that may lead to the drug development. So I think many of you are the research scholars, research students or pharma students. So as you know, in India, there are lots of research centers and universities. They're working in many research activities. So whether all these research led to the development of drug, we have the source. As you know, in India, it is a very unique in biodiversity. We have a different agroclimatic zones, and the Western Ghats, it's a very rich heritage or rich source of many valuable medicinal plants. So whether all the research lead to the development of drug or the development of a specific treatment modality, that's a question. If you search anti-cancer potential of a medicinal plant in Google or in Scopus scientific base, you might have seen thousands of research articles with anti-cancer activity or anti-diabetic activity. So there are many research happening in India and even globally. So what will the final outcome that will be directly reach the society for relating any disease conditions? So actually what happened in the research field, academic and or industry field, what actually activities. If it is a PhD scholar, finally they have to submit the thesis and to publish some articles. So that will be the preference. But if you are doing a proper research, the PhD thesis is only the starting of the research. So finally, normally while presenting the thesis, there should be a last slide, future perspective. That should be the only in slide only. There is no more research apart from the student or from the mentor or guide. So research should be very focused. Any research outcome should be definitely, finally it should be reached to society. So if you are doing any anti-cancer activity of medicinal plant or anti-diabetic activity of medicinal plants, we can publish some research article, we can get some funding from research funding agencies. Apart from that, it should be benefited to our society. So that will be the research focus. So I'm focusing on some traditional validation of knowledge base, how it can be converted into a development of new drug. So as you know, the traditional medicine, now it is very boosting in the, specifically in the herbal medicine industry. And during the last few years, we have in the pandemic situation of COVID-19. So that time also, there is a boost for natural product to traditional medicines. And it is expected to reach the herbal medicine industry over the globe around $550 billion by 2030. So there are different products like, apart from the medicines, there are nutraceuticals, cosmetics, flavors and fragrance, and also natural additives, natural preservatives. So these industries enlarging, enhancing due to demand in medicinal plants and herbal medicines. At the same time, there is another phase for this. There is a scarcity of medicinal plants because, as you know, the medicinal plants containing a variety of chemical constituents. Even a single medicinal plant contains maybe 10,000 to 20,000 of compound. There is normally there's a question, if anybody is doing the research work in the medicinal plant or phytochemistry, normally students are asking, sir, I want a plant which is not yet explored or not yet studied for getting a new compounds or new activity. See, as you know, the common example like Osimum, Tulasi, it's a very common studied plant. Even after 100 years, anybody can isolate a new compound from the same tulsi. Because a single plant, a part of a plant, so for example, leaf of a tulsi contains thousands of compounds. Maybe if you just search the literature, maybe some hundreds of compounds have been reported. If it is a well-studied plant, even it contains a large number of compounds. So it is a huge reservoir for many active principles of medicinal plant. And we have, as far as our country is concerned, we have a Western Ghats, and also there are biodiversities very specific and unique for our country. So still, the search for the various drugs for specific disease can be explored using these medicinal plants. So let us see how a traditional knowledge base can be translated into drug development. So we have a various traditional knowledge base like Ayurveda. The Ayurveda classic texts have been written around 5,000 years back. So there are many descriptions regarding the specific medicinal plants used for a specific set of conditions. That will be the starting point for a drug development from the traditional knowledge base. So we can extract these medicinal plants for getting the desired type of compounds using various solvents, various combinations. Different extract of the same plant or extract of combination of various medicinal plants that can be properly studied to identify the desirable compounds using various phytochemical techniques, mainly like chromatographic and spectroscopic techniques. Once we identify the metabolites from the plants, it can be good for the validation. If you are focusing on a specific set of disease condition, that can be done in vitro model, or once the metabolites are identified, we can use in silico model, then in vivo model, and once it is get established its efficacy, definitely you have to go for the toxicity studies. Whether any toxic effects are there. I think the pointer is working here. Okay, fine. So for that there is some one problem is also there. Normally the Ayurveda and herbal drug industry is facing the shortage of the many medicinal plants. So scarcity of medicinal plant is the most important problem faced by the any herbal drug industry. So many valuable medicinal plants are become endangered or extinct. So that will lead to the adulteration or unauthorized substitution. That is a very important thing, because any formulation, if the raw drug is not properly identified, that will affect the quality and efficacy of any formulation or any drug. Suppose sometimes, for example, if you are identifying a wrong identity of a medicinal plant, if an allied species or similar species, it may not be harmful, but it will not be efficacious. So the quality standardization is the most important thing. Whenever you speak the drug development from traditional knowledge base or herbal medicines, the stringent quality test is required. So for the herbal medicine or the plant origin, there are different stages of quality standardization, like taxonomic using a botanical characters or morphological characters, then anatomical using the microscopic characters, phytochemical, and also DNA based molecular fingerprinting. So quality standardization is the important thing. So we can identify the genuinity or authenticity of a medicinal plant or a herbal plant material using various techniques like taxonomical, phytochemical, and molecular techniques. I'm just showing one example. This is our own software database developed by our institute with CSCR Series. It's a herbal authentication system where the anatomical or pharmacological features have been stored in the database with the genuine plant which have been collected from our own herb garden. So once we are getting the samples from outside, we will compare the characteristics using this software. So we can identify whether the sample in the market or in the testing sample is genuine or not. So using these anatomical characters, we can identify the quality and also the authenticity of the medicinal plant. And the second one is the phytochemical. As you know, normally every medicinal plant containing a defined spectrum of phytochemicals. So using various chromatographic and spectroscopic techniques, we can store a phytochemical database using chromatographic techniques like HPTLC, HPLC.
gas chromatography, and spectroscopic techniques like FTIR, mass spectrum. So when you are getting the sample, you can extract the same method and identify the fingerprint whether it is matching or not. So these are the normal techniques using for the quality standardization as far as the herbal medicine is or medicinal plant is concerned. So as you know, in the case of herbal drug industry, only few items are used in a fresh form. Most of the raw drugs are being used in a dried and chopped form. So it is quite difficult to identify the authenticity using the physical characteristics or morphological characteristics. See, for example, the bark of Sarakashoga. Sarakashoga is an important medicinal plant used in Ayurveda, particularly for the estrogenic activities. So it is the most adulterated species in India. So many bark of other trees also being sold as Sarakashoga. Morphologically, it will be very similar to Ashoka, cannot be distinguished using the physical appearance, but we can use the other techniques like anatomical or phytochemical techniques to distinguish which is the adulteranol and which is the genuine one. I will show one example. One is the Sarakashoga itself. As per a survey conducted by National Medicinal Plant Board in India, 80% of the Sarakashoga is adulterated in Indian herbal drug market. So if 100 kilogram bark of Sarakashoga available in India, only 20 kg is genuine one and 80 kilogram is adulteranol. So it is a very, the adulteration is its magnification is very huge in the case of Sarakashoga, but it is a very important Ayurvedic medicinal plant also. So let us see what is the macro and micro characteristics. See, this is the genuine Sarakashoga, and this is the Polyalthia longifolia is the common adulterant, which is in Malayalam it is known as Aranamara. This is the most common adulterant for the, and sometimes even sometimes it is misunderstanding whether it is Polyalthia or Sarakashoga. Even some houses also they are growing the Polyalthia as Sarakashoga, but actually it is a different plant. And there are other like Trama orientalis, Shoria robusta also being used as Sarakashoga. And see the chemical profile also. This is HPTLC profile. The first one is the genuine Sarakashoga. All others are the adulterant, which are physically very similar. We cannot distinguish which is the Sarakashoga or other species, but chemically it is totally different. This is HPTLC profile. In HPLC also the adulterant is totally different, and LCMS profiling also the compounds are totally different. The Sarakashoga contain isoflavonoids and phytoestrogens in higher quantities. All other plants are that compounds are not present, even not present in the other adulterant plants. This is the molecular based, DNA based identification. Here also there is one advantages for the molecular identification because it is free from the environmental or other agroclimatic conditions variation. Sometimes say in the phytochemical, the chemical concentration of the individual chemical compounds may differ according to the agroclimatic zone where the plant is grown. But being in molecular, it is based on DNA, genetic material, it will be very stringent method for the identification of a raw material. And this is another example. It is a Tragia involucrata. This is another most adulterated species in raw drug industry. It is in Malayalam it is known as Kodithuva or Choriyana. I think you know that it's if that leaf is touched with our body, it will get some dermatitis and irritation. So this is also see the different in the chemical constituent in the adulterant and original species. The black one, the HPLC profile, this is the genuine sample, and the blue one it is adulterant. The chemical profiles are totally different. The major chemical constants are not present in the adulterant. Another example is the cinnamon, because it is being used as spices also. The cinnamon xylanicum is the genuine species, but there is another species which is known as cassia cinnamon, which is very cheaper material. So in the spices and food industry, this is being traded as cinnamon, but which contain a toxic compound coumarin. That's why FSSI, there is some regulations. The maximum content of coumarin is not more than 0.3%. So this is a market survey. The sample 1 and 2 are collected from the spices market of Malappuram district. And the third one is the genuine cinnamon xylanicum collected from our herb garden. You can see that the market sample containing high quantity of coumarin, but the genuine sample is free from coumarin. So it will be more cautious. Nowadays there is a problem. If we are using in a food, we are using only a little quantity as a spices. But there is a practice now for anti-diabetics. The people are using as a some Ottamuli treatment, the powdered cinnamon being consumed for the diabetes. So if it is in a powdered form, the absorption of compound will be more. So if you are using a adulterant, it will be very toxic for liver and kidney. And another common adulteration is the citrus deodora. It is Devadari. It is very costly material. So in this case, there are two types of adulteration. One is a totally adulterant species, and another one is the plant. It is, as you know, it contains many volatile essential oil, and it is being used in the perfume industry also. So what happening? The plant, it is being extracted the oil separately. So the leftover material being traded to road drug industry, and oil will be traded separately. So this is on market sample. You can see that the first one and second one, these are the citrus deodora, but it is in different qualities. You can see that the second track which contain the higher abundance of the compounds, and the third one is the oil extracted sample. The same citrus deodora, but it is a oil extracted sample, and the fourth one is a totally adulterant. And now the question is, as far as the Ayurveda industry is concerned, all the preparations or decoctions are being prepared in water. So citrus deodorant is being boiled in water at 100 degrees Celsius. So it contains volatile oils, but definitely it will evaporate. So there is a scientific question whether Ayurveda industry required a genuine sample, because if you are preparing the formulation using water, the volatile oils will be evaporated. So even you can use the leftover material. But we conducted a study preparing the decoctions with two materials. One is with a genuine Sarakashoga, another one is we prepared decoction with the oil extracted sample. But the profile was totally different. So even if you are preparing the decoctions in water, it should be the proper citrus deodora with the oil content, because certain compounds are still existing in the formulation even after boiling with water. So the first part is regarding the quality standardization. Now let us move to the second part, that is herbal drug development from traditional knowledge base. So as you know that validation of traditional knowledge base using the scientific techniques, that will lead to the many new drug or new formulation. So as we know that there are many phytochemicals originated from the plant origin. So for example, reserpine, I know you might have known about the reserpine. Just a compound isolated from Rauwolfia serpentina. It was used as an anti-hypertensive drug, but now it is not in the market. It has been banned due to its adverse effect. But Rauwolfia serpentina, it is still used in Ayurveda. In the Sarpagandhadi Churna, it is used for anti-hypertension. There is no toxicity at all. So while if you are using the reserpine as a single drug, it is showing many toxic effects. But in Sarpagandhadi Churna, which contain along with the Rauwolfia serpentina, many other medicinal plants are also there, and which also contain the same compound reserpine. So that is the holistic approach of Ayurveda. So all the preparation, or there are certain single drugs also there, but that is also a single medicinal plant which contain many phytochemical compounds. So the mechanism of action of Ayurvedic formulation is it's a multi-component, multi-target, and it's a polypharmacology. There is no single drug for a specific disease. If you, for example, if you are, if you have some fever, if you approach a modern medicine practitioner, modern physician, if 4 persons together consulting a modern medicine practitioners, definitely one medicine will be common. It should be paracetamol or mefenamic acid. But if the same 4 persons is visiting an Ayurveda physician, sometimes there should not be no common medicine. The physician prescribes the medicine according to your body nature, physiology, in their termed as a prakriti. So why these formulations are acting? See, if single formulation containing different compounds, and in the case of Ayurveda formulation, the compounds having the structural similarity, at the same time there are some functional similarities out there. So on kashaya or on decoction, the physician has to be decided at what condition it should be prescribed for a patient, depends upon the natural physiology of that patient. Because that multi-component, that multi-target compounds will act specifically according to the physiology of that body or that patient. So say for example, one formulation containing 10 compounds, but in a specific patient, only few of the compounds will be acting, that will be binding with a specific target. So that target or that disease condition should be defined by the physician. But now there is no practice like that. Say any Arishta Kasha which is available in many outlets of Ayurveda industry. So people are consuming. For example, during the COVID-19 time, lockdown period, the most wanted medicine, Ayurveda medicine was Arishtas. Because due to lack of some other Arishtas, people are used to the Arishtas without the prescription of any physician. So what happened? So if you are consuming continuously 3 to 4 months a high dose Arishta, definitely your liver will be damaged. But all the campaign will be, allegation should be against the Ayurveda. No Ayurveda physician will prescribe Arishtas for a overdosage for many months or weeks. Should be a dose optimizer. So now there is a propaganda regarding the toxicity of Ayurvedic medicine. As you know, being the pharmacy student, in the universe everything is toxic only. If you are drinking water more than 7 liters per day, it will be toxic. So everything is toxic, only thing is the dose. If you are using any medicine, system of medicine, whether it is modern medicine or Ayurveda, anything, it should be in a specific. There is a dose will be there. So this is the holistic approach of Ayurveda. So I am focusing on, as you know, there are many, like Reserpine, there are many compounds. But the only thing is, if you are focusing on a single compound, you do a stringent toxicity studies and pharmacological studies. Normally I never recommend the students for doing a PhD work for, I will recommend for isolation of compounds, but normally while they are submitting the synopsis, isolation, characterization and anti-cancer activity of the isolated compound. It is too ambitious. Suppose if you start with a 10 kilogram of raw material on plant material, if you are isolating a compound, finally you will get at least 20 mg or 30 mg. 10 mg or 15 mg will go for the NMR studies to identify the structure. Then remaining 10 mg or 5 mg, you cannot do the analysis, all these things, toxicity studies and other things. So if you are focusing a single compound, and finally if you are getting a good activity, finally if you go for the toxicity, even in the acute and subacute toxicity studies, it will be toxic in nature. In many cases that is happening. So that's why even pharmaceutical industry also converting, that is, as you know, the many compounds are there in the medicinal plant, but it is quite difficult to commercialize these compounds. Because of all these compounds in the plant, all these compounds are not in a pure nature. So isolation itself is a very task. Once it is isolated, you have to isolate in a commercial scale, it is quite difficult. And also, stability is also another problem. As you know, if you are doing any research work, we can get the standard from Sigma Chemicals. It's very costly. Once it is opened, we can keep at least 5 days or 6 days with a stable condition. After that, if you spot in HPTLC, it will get, instead of single band, it will get multiple bands. That means it get degraded. So now the focus is regarding the standardized herbal extract or poly-herbal formulations which contain multiple ingredients of different medicinal plants. So there is an integrative, now globally there is an integrative approach for specifically for the cancer treatment, management of cancer. Along with the conventional cancer treatment, there are certain standardized extract or traditional medicines along with the some lifestyle modification, diet modification that will improve the quality of life of a cancer patient. So most important thing is, as you know, even after the chemo or RT, there should be some adverse effect for the patient. Definitely in case of the cancer treatment, you have to opt the modern medicine because it's a very advanced and evidence-based treatments are there in the modern medicine. So that can, the traditional medicines can be a complementary therapy for the management of cancer. As you know, we have our own experience. We are conducting at our charitable hospital, there is a cancer outpatient department is there. We are not claiming the cure for cancer, but we are treating the cancer patients for improving the quality of life. During the last, just 2 years back, we have just analyzed the data of the cancer patient visited the last 10 years in our cancer OP clinic. It was more than 10,000 patients. It is very comparatively, it is a high number, even comparing with a modern oncology hospital. So we just analyze the data of the patient. So most of the cases, the patients improved their quality of life with certain herbal formulations. Specifically in the case of CA breast and CA lung. So even in the case of CA lung, if it is a EGFR negative mutation, there is only a limited options are there in the even conventional treatment. So in such cases also, normally it is a 7 month is a prognosis, but in our history, it is more than 21 months survived the cancer patient with a 4-stage CA lung patient with EGFR negative mutation. So it is improving the quality of life. At the same time, it also increase the survival time. So integrative oncology, now the topic is the very hot topic in the discussion in the cancer management. Many countries like Japan, they have also included, also Korea also, they have included the complementary therapy of traditional medicines along with the conventional treatment. That will be a better idea for the management of cancer. Now, normally there is a question whether any regulatory guidelines are there for the herbal medicine. Definitely, for the Ayush systems of medicine, there are different regulations. This is one roadmap for the regulatory affairs regarding starting from the 1962 Pharmacopoeia Committee, and finally we have the separate ministry for Ayush. And Ayurveda Pharmacopoeia of India and Ayurveda Formulary of India. These are the national documents for the prescribing the quality of the single drug and also the formulation. So all the Ayurveda drugs or herbal medicine should be qualified or tested for these parameters as per the standards mentioned in the Ayurveda Pharmacopoeia of India. Now let us see what is the, I have mentioned that there is a polypharmacy or polypharmacology of a polyherbal formulation. As you know, many of the Ayurveda preparations are polyherbal in nature. So there is a chemistry acting behind the polyherbal formulation. Say for example, any decoction or any kashaya which contain more than 2 or 3 plants, certain formulation contain more than hundreds of ingredients. So whether all these ingredients are required for a formulation or what are the compounds separated in the preparation of this formulation. So in the polyherbal formulation, there are 2 chemical processes happening. One is the synergism and another one is the antagonism. Synergism, simply speaking, it is in synergism, 1 plus 1 is not equal to 2. Sometimes it may be more than 2, sometimes it may be less than 2. And antagonism, as we just cited the example of reserpine and sarpagandhadi churna. Reserpine is banned due to toxic effect as an antihypertensive drug. But still in Ayurveda, sarpagandhadi churna is being given for the patient which contain Raulfia serpentina that contains serpine. We have done the profiling of sarpagandhadi churna which contains the serpine. But that reserpine not causing the toxicity. That is antagonism. The toxic effect of certain compounds will be nullified by other compounds. So these are the 2 chemical process transformations happening in the formulation. And so in the case of a polyherbal preparation, all the steps, if we are preparing a formulation with the proper method, it will be very efficacious and you will never show any toxic effect. So starting from the ingredients, so we have to first authenticity of the raw material, then quality of the ingredient plants, whether it's a good quality or a bad quality material, then method of preparation is also very important. There are specific methods mentioned in the Ayurveda text and also the Ayurveda Formulary of India that should be followed properly. So up to the final drug, there are different process which should be very scientific and properly prepared. Then only it will be a correct formulation. This is one example of such study we have conducted in our lab. It's on Amrotram Kashaya, Ayurveda people can easily understand. So which contain 3 ingredients. One is the Tinospora codifolia, our chittamarde, then terminalia cebula, kaduka, and gingiber officinally, dry ginger. These are the 3 ingredients of Amrotram Kashaya. So we have done a scientific study in comparison with the Amrotram Kashaya prepared as per the AFI, and all these ingredient plants are being prepared singly. So we have the 4 samples, one is Amrotram Kashaya, one is the Kashayam of gingiber officinally, another is Tinospora codifolia and terminalia cebula. We just compare the chemical profile individually along with the formulation. So the chemical profile of individual plants and formulation is totally different. So we see the spectrum of these formulations. You can see that this is the final formulation and this is the ingredient plants, 3 ingredient plants. There are many chemical shifting is happening while it is in the formulation, particularly for in the case of gingiber officinale. Many compounds present in this gingiber of fission being converted in here, and certain compounds present in the terminal cebula and tinospora codifer that are present as such in the formulation. So we have conducted detailed studies using the mass spectrum analysis. So we can see that here in the individual extract of gingiber of fission, these are the major compounds present in the gingiber of fission. But see, in the formulation, only 3 compounds are present in the final formulation. So if you are preparing a decoction of gingiber separately, it contains more than 10 compounds, but out of these only 3 compounds are in the formulation. So we just removed gingiber from the system. We prepared a decoction using terminal cebula and tinospora codifolia. The chemical profile was totally different. In the absence of gingiber, many compounds from tinospora and terminalia was not present in the formulation. So gingiber is a bioenhancer. As such, it is contributing only 3 compounds, but it enhances the extraction of many other compounds from the other ingredient plants. That is in the Ayurveda, they are taking like a yoga, one specific yoga. So if you are taking a specific yoga, all the ingredients should be present. So any formulation which is prepared out of 10 ingredients, 10 should be there. If it is prepared by 9 ingredients, it will be different. So that is the synergism and antagonism happening in the polyherbal formulation. So you can see this is available in, this article is available. See, actually this article is published in the Journal of Applied Spectroscopy. It is not anywhere related to Ayurveda or herbal formulation. It's only a journal which is dealing with the physics and chemistry. While I communicated this journal, this article, the editor itself asked why you are communicating this article into Journal of Applied Spectroscopy. Many Ayurveda journals are available. Because being Amrut and Kashayam is an Ayurveda, the science is only chemistry. The spectral changes, chemical transformation. So it will took 2 years to publish this article because many queries are there from the foreign reviews. They don't have the idea about the polyherbal formulation or synergism and antagonism. But we purposefully published this article because if you are publishing the article in the Ayurveda journal, only readers will be in the Ayurveda community. No other scientific community will realize what happening actually in the Ayurveda field. So these are the chemical changes or scientific basis of our traditional knowledge base. Only thing we have to identify or we have to validate using the modern techniques. So see, this is why I showed all these things, that you have to realize the holistic approach of the Ayurveda. So that is the peculiarity of polyherbal formulation. So polyherbal formulations are, that's why see, for example, Chavanaprasha, we are using thousands of years Chavanaprasha. So for classical Ayurveda, normally there are 2 types of products are there in Ayurveda. Only is a classical product as per the Ayurveda classical text. There is no toxicity studies or anything is required. We need not to establish the efficacy of Chavanaprasha now. We can do as a research purpose. Even without the data, we can prescribe or we can consume the Chavanaprasha because it's a time tested. For the thousands of years, it is being in the practice. But if anybody is preparing with some spurious material, definitely it will cause toxicity. But the problem is not with the Ayurveda, it is problem with the manufacturer or the vendors of that product. If it is not properly prepared, definitely it will cause toxicity. In any medicine system, whether it is modern medicine or any other system, definitely will cause toxicity if it is not properly prepared. So now there is another approach regarding traditional knowledge-based metabolomics approach. I think you pharma students know about the metabolomics. So identifying the metabolites from the lead of the traditional knowledge base. Say for example, in Ayurveda there is some drug like Medhya Rasayana, which will improve the intellect. So there are many medicinal plants prescribed as Medhya Rasayana. So we have to validate as scientifically. So what we are doing, as a scientist we are doing, it will be identifying all the metabolites from the Medhya Rasayana. What will be the nature of compounds which are present in that plant. And any common compounds are present in all these plants. In that cases, the similarity is in 2 ways, only structurally similar, another is functionally similar. As you know, in the case of modern medicine also, if you are using the omeprazole or rabiprazole or esomeprazole, everything, anyway, it will reduce the gastric irritants. The structures are different but the same act. The same thing, many phytochemicals having the similar structure but have the different action, or different structure have the common properties. So that should be also we have to consider while comparing the phytochemistry of a medicinal plant. So normally for the metabolic profiling we are using the LCMS, liquid chromatography mass spectroscopy for the metabolite analysis. That is a very fantastic tool for the metabolomic analysis. It is being used in the different field of branch of science like forensic department, pharma industry, natural product industry. There are different applications are there. So normally liquid chromatography, Q-TOF mass spectroscopy, quadrupole time of flight mass spectroscopy, it will give the accurate mass spectrum that will lead to the identification of compounds. So using a LC-MS spectrum, we can identify the metabolites in a medicinal plant that can be compared. Normally, with a single mass value, we cannot, as you know, all the mass spectrum give the M by Z value, mass by charge ratio. So if you are getting only the mass value, see for example, if you search a mass for 170, thousands of compounds will be there. So if you are doing in a Q-TOF LC-MS, the mass will be 170.1234. Up to 4 decimal, it will be accurate. And we are doing the mass fragmentation. Once the compound is fragmented, comparing the fragment, we can identify properly the compound. So using that technique, we can identify the metabolites. If you are doing a LC-MS spectrum of a single plant, it gives the thousands of compounds. By using the MS-MS spectrum, we can identify at least 250 or 300 compounds from that profile. So then using the chemometric software, we can identify which are the common compounds present in a plant, in a different extract, and what are the structurally similar compounds in various plants which are being prescribed in traditional medicine for a specific set of disease. So this is one approach for the metabolomic basis of the drug development. Now I just showing a few examples how drugs are being developed using this metabolic profiling. This is on proprietary medicine. Apart from classical drug, there is on another area, patent and proprietary medicine. These medicines should be prepared using the medicinal plants which are mentioned in the classical Ayurveda text. It should be mentioned in the classical text, but not in a specific combination. So this is a new drug developed by Ayurvedishala. It's a Mensoko tablet which is being used for the menopausal diseases and some gynecological issues like uterine bleeding. So this is based on the validation of traditional knowledge. There are many medicinal plants which are mentioned in the classical text for the improving the uterine health. So we just identify the many compounds from the set of plants and we develop a drug. So the final drug which contain the phytoestrogens which can mimic the activity of human estrogen. So in the clinical trial, from the physician feedback, it is very efficient for the uterine bleeding and some other gynecological issues. So the scientific study showed that which contain the phytoestrogens, the active phytoestrogens are present in the formulation. So this is not a classical formulation. We developed a formulation based on the traditional need of Ayurveda. So this is the article regarding that which is published in a gynecological journal, Asian Research Journal of Reproduction by elsewhere. This is another example. As I already mentioned that in our cancer OP, we have been given some specific formulations instead of combination in the cancer patient. And we are from the feedback, patient feedback, it is, see these are the one formulation. Actually it was developed by the suggestion from our forum managing trustee, Dr. P.K. Warrior, late Dr. P.K. Warrior. Because his clinical experience, he only suggested to combine this formulation and develop a new drug, KGS tablet, which contain the 3 combination, that is cyanidin, daclyon and cyanidin scenario. So, which contain specific compounds which can prevent the growth of breast cancer cell lines. So, all these medicinal plants are mentioned in the classical Ayurveda text, but we developed a drug based on that traditional lead using the scientific validation and from the clinical practice and clinical feedback. So this is the article published in the Natural Product Research. It is published from UK by Taylor and Francis. And this is another example. Now this is in the pipeline. We have already signed an agreement with the Center for Cellular and Molecular Biology, CCMB, Hyderabad, for developing a herbal drug for neurodegenerative disease. That is the most important problem, particularly in case of Kerala, because our lifespan is increasing. So geriatric people are increasing. So there are many problems regarding the dementia and Alzheimer's. The cases are increasing. And even in the modern medicine also, there's only limited options are there to manage the neurodegenerative disease. But in Ayurveda, there are many medicinal plants have been prescribed like Rasayana drugs or major Rasayana drugs. This one plant, Ipomia mauritiana, which is termed as a Ksheera Vidari in Ayurveda, which is termed as major essence, which will improve the intellect as per the traditional knowledge base. So we have done the anti-amnestic activity in animal model. So we have identified this as a specific extract. We have optimized the method and we identified a active extract which improve the quality of the intellect of the patient. So now we are trying to develop a herbal drug using this plant along with other combination in association with CCMB Hyderabad. So all these are I'm showing, these are the example how traditional knowledge base can be converted into lead to the development of a new drug. This is another example. There is two plants, Duptiga and Leguduptiga, that is Euphorbia thymifolia and Euphorbia hirta, which are mentioned in the classical Ayurvedic text. We just compared the metabolites. So both these plants contain similar type of compound. So we have done the anti-cancer activity of these plants. So we show the potent anti-cancer activity comparing to the chemo drug 5-fluorouracil. And this also now we are collaborating with the Cancer Biology Division of CCMB, and we are also trying to develop a specific drug for cancer treatment. And now, apart from the drug development, whether we can develop a new treatment modality from the traditional knowledge base. This is one example. We recently collaborated with the University of Johannesburg, South Africa. There is a laser research center where they are using the laser photodynamic therapy. That is a laser type of treatment for the cancer patients. Normally, they are in practice with a synthetic drug, investing in synthetic drug, applying with a suitable wavelength of laser. It will be release the reactive oxygen species that will kill the cancer cells. But in the synthetic drug, there is some adverse effect. It will kill the normal cells also. So this is the principle of photodynamic therapy. There is a photosensitizer, that means any compound that can be converted into energetic state by absorption of a laser beam with a suitable wavelength. That is the principle of photodynamic therapy. Now whether this technique can be utilized using our traditional knowledge base. So we have selected a certain medicinal plant extract which also contain some photosensitizing compound that can also be used as for photodynamic therapy. So if it is success in certain type of cancer, specifically like a skin cancer, so there will be a new treatment modality, the photodynamic therapy of modern medicine system that can be utilized using our medicinal plants also. So it is the work is under progress with the collaboration of Johannesburg University. And now the question is whether anything can be done in the case of pandemic like a COVID for the traditional medicinal plants. So before presenting all this, let me to say that there are many other fake claims also there on behalf of the Ayurveda and traditional medicine. But if you are doing scientifically, there should be some provisions in the traditional knowledge base for any diseases. We have seen that even the modern medicine repurposing of drug for a set of new disease. The same thing we have done in our institute also, repurposing the traditional knowledge base. This is one product which has been released by Ministry of Ayush itself, Ayush Koda Churna. Maybe you will be familiar with this drug. Still it is in the market. Which contain, it is a powdered form, it is a Churna powdered form of Osimum tenuforium, cinnamon verum, gingiber officinalis, piper nigrum. So we just analyzed the chemical profile of this drug, whether it contains any promising molecule. So from our study, it found that it contains many immunomodulatory and anti-inflammatory compounds. So from the patient feedback, the moderate symptomatic patient, and our clinic also it is being given to the COVID patient. So it has showing some relief from the other symptoms, particularly for the post-COVID symptom like pneumonia like or the chest congestion like that, to be getting relieved by those patients having their consumed the Ayush Koda Churna. So which contain many compounds which having the immunomodulatory and anti-inflammatory property. And also three other classical formulation also we have used for the repurposing. One is Indugandham Kodam, another one is a Villadi Guliga, and third one is a Mukkamukudvadi Guliga. We just analyzed the chemical profiles and identified the chemical compound, and we found that the Villadi Guliga contain many antiviral compounds, not specifically for COVID, but many antiviral compounds. And Indugandham Kodam, which contains immunomodulatory compounds, and Mukkamukudvadi Guliga is an anti-inflammatory compound. So These three medicines are being given for the COVID patient during that COVID season in our clinic. So in the case of Bilwadi Guliga, it is getting the symptom is get reduced even in 5 or 6 days. So we just converted the dosage form of the Bilwadi Guliga. We just changed its formulation, and we have done the study, antiviral study, specifically in SARS coronavirus 2 in collaboration with the CCMB Hyderabad. So we found that the Bilwadi Guliga extract can reduce the viral load even up to the 50%. So this is a classical formulation which is already in the practice in Ayurveda, but that drug can reduce the SARS coronavirus. It has been tested in the 3 strains which are available during that time. So it can reduce the viral load in the patient. So this article published regarding that research, which has been featured by WHO and NIH also. This open access journal is available in the scientific basis. So based on that traditional lead, we have signed an agreement with the CCMB to developing a herbal drug as an antiviral drug for SARS coronavirus 2. But still, we have started the study in 2020, but still it is in running. That's why I'm showing if you are doing the proper scientific research, it will take time. So any herbal drug or COVID-19 drug which are being claimed so as without any proper study, so that is not a correct scientific way, but still this is almost in the final stage. Now the toxicity studies and dosage fixation is going on. But the problem is, even by changing the every strain of COVID virus, the IC50 value is changing because it is acting like as every variants in the COVID-19, it will be different. It is differently acting against this drug. That is good. Many times you have to repeat around 6 or 7 times to get the proper IC50 values because being it is changed the variance, mutant variation. So let me conclude the presentation. So I just showed how the traditional knowledge base can be converted into new drug development or developing a new treatment modalities. So as far as our country is concerned, we have a long traditional knowledge base used. As you know, we are in practice in Ayurveda and many other traditional systems of medicine for thousands of years. We have a large reserve of medicinal plants, and we have the Western Ghats with enormous potential of medicinal plants still unexplored. So proper scientific studies may lead to the development of new formulations or new combination for the management of various disease. Only things we have to do the stringent scientific studies to ensure the therapeutic efficacy and most important the safety. So if you are using the spurious material, if you are, as I mentioned in my earlier slide, if you are identifying, if the raw material is wrongly identified, that will definitely affect the quality and efficacy. Sometimes it will cause toxicity also, if you are not properly identified. So starting from the identified raw material with the good quality, that's the most important thing. So using that traditional knowledge base, we can improve the clinical practices, we can improve the treatment modalities, and also we can develop a new drug, particularly in the case of a polyherbal formulation and the standardized extract that can be used for many diseases without any toxic effect due to the synergetic and antagonistic reaction, which are all these formulations are in the polycomponent multi-target with the polypharmacology. That means even one formulation can be used for the different diseases. Only thing that should be prescribed by a proper physician. Okay. So I acknowledge to my research team. They are the real heroes for all the studies. So thank you very much. Anything regarding the topic, you can ask many questions. I think definitely 5 or 6 questions is awaited. Okay, thank you. More than that. Okay, definitely, sure, sir.
Sir, whether we will allow our students project because LCMS is all here, and students are doing.
Yes. We have the program for short-term training and MSc project. So please visit our website. The procedure is there. We are allowing the students, we are taking the students based on their merit only, based on the UG and PG first year marks. Yes, you can apply for that also. Sure, sir, you can apply. We have the program of 1 month to 6 month training program For project, 3 months is compulsory. Yeah, apply online. You can application forms are there in the website. You can download and apply it by mail. There is an email ID is given in the website. You can send to the application to project director. Please note the website cmbravs.in. It is there in the website. How to apply for the project or training. Thank you, sir. No, see, if it is a singly isolated compound, definitely HNMR and correlated spectroscopy is required. All these compounds are, it is already reported compound, and we are using LCMS, FTIR, UV, and HPLC, HPTLC together to identify the compounds. So in our instrument, we are doing the MS and MSMS, and using the mass professional profiling. This is a software which is provided by Agilent itself. So we are selecting the compounds which will match more than 95% score with the mass match. So we are considering the empirical formula, molecular formula, and mass match, along with the retention time and other parameters like FT and UV structure. So we can confirm the structure. Yes, yes, correctly. Actually, we have done some validation of some purification studies. So in that, in between, there are some byproducts also being formed, but finally it will get nullified. Thank you. I think some questions from students area also.
Infections are a very common problem among oncology patients. So often
Please louder, I am not. Please.
A very common problem among oncology patients.
I just missed your first word. Inflammation. Infection, yes, please.
Problem among oncology antibiotic or even maybe.
I think that cordless mic range is not much there, so you can just come forward and ask. Okay, it's fine. Yes, correct. That is a good question. As far as the cancer patient is concerned, the infection is an important problem that faces. Particularly, if any infections occurred, even my antibiotics will not work. So, in our physician also, there is some history. Please, you can see, in certain cases, some cancer patients occurred some infection, and it is not responding to any antibiotics. So what happened? The physician suggested to do some purification process, like Panchakarma treatments are there, like Vasti, some other treatment. So they have done some Panchakarma treatment. After that, they are given the same antibiotics. It responded very significantly. So now the problem, even also now there is some research program by ICMR, Indian Council for Medical Research, regarding the antibiotic resistance. During the COVID time, because there is one the bad practice of our media or even social media also. See, azithromycin, even my child studying in 5th standard know azithromycin. Because any sore throat, something happening, simply buying the azithromycin and consuming. So now what happened? It's not at all affecting. Amoxicillin, azithromycin, cefuroxime, anything not responding to the strains. So that is a bad practice of physician also. Normally in any other countries, without a prescription, nobody will sell the antibiotics. But even now, anybody can, even without any dose, if you want 50 numbers of amoxicillin, you will get the 50 numbers of amoxicillin. So what happened? The antibiotics are not responding at all. So even as you know, the flu, common flu, now it is a season for common flu. Everybody knows it is a viral disease. There is no role for antibiotics. If any secondary infections are there in the throat, it can be suppressed by simply saline gargling. But we will start the high dose antibiotic initially. So that is a bad practice. Now even the modern physicians are also aware about that. ICMR strictly, even WHO also don't prescribe any antibiotics for the viral diseases. Now I think most of the physicians are. So in Ayurveda can do like this. If any antibiotics are not responding, they will do some treatment for the body purification, like a Panchakarma treatment. So once the Panchakarma treatment is over, the body will be more susceptible to our immune activity. And regarding immunity also, there is some like that. We are boosting the immunity. There is no boosting of immunity. We cannot boost immunity. Immunoregulatory only. Every time we need to the enhanced immunity. Sometimes it should be suppressed. So body automatically will regulate whether need to enhance or suppress. Some allergy condition should be suppressed, not to be enhanced. So it is immunomodulatory, not immunobooster. There are many even articles and scientific articles are there. Immunity boosting, immunity boosting. So actually it is immunomodulatory only, but it is misspelled normally. Immune booster, not immune booster, immune regulator only, or immune modulator. Yes. Yes, there are immunomodulated medicines are there, but that should be prescribed by a physician. I think our chairperson can say more about that. Because there is no common immunomodulatory drug like that. There is not. So Indugandham Kodam, it is normally common as immunomodulator, but that also should be consumed after prescription of it. A physician can prescribe or optimize the dose for. There is no it is in every system there is no common medicine. Now the pharmacovigilance are stringent by the Ayush also. I know there are certain advertisements are there, even in the brand of Ayurveda, for improving the there is no such medicines are there. Whether you have seen any advertisement like that to elevate the cough and something like that, any advertisement of amoxicillin or azithromycin, not legal for Ayurveda also. For a specific set of disease condition, you cannot advertise any medicine because that should be prescribed a physician only. You have the infectious cough. Whether you have to go for amoxicillin or azithromycin or any other antibiotic should be prescribed by a physician who examined you physically. But nowadays, even a pharmacist can prescribe the medicine. Many times, even I have overheard about any person come to a pharmacy, I want medicine for cough, toothache, stomachache, and everything will be given to. So that's why the antibiotic resistance is more prevalent nowadays, due to the misuse of antibiotics. And there is another practice also. Physician prescribe antibiotics. And there is one question in the pharmacy, the people, for how many days? 5 days. Then they will say for 2 days. So after 2 days, they will stop the antibiotic. So what will happen? The strain will be more susceptible for that strain. So next time, it will not respond to the same antibiotic. That's also another problem. Missing the dose form. So Ayurveda can enhance such condition using some Panchakarma therapies.
In Ayurveda, the antibiotic, instead of antibiotic, they use basma.
Yeah, that is it. Not like that. That's also specifically according to nature of the disease, age, many things are, sir. Now there is one most important scientific research regarding epigenetics. I think some of you heard about epigenetics. As you know, our genetic material DNA, but due to our environmental condition or food habit, that can be altered. Some DNA methylation or chromatin aberration. That can be transferred into your other progeny also. So normally, Ayurveda is considered as an ancient science of epigenetics. So many of your formulation can be capable for reversing these changes in DNA. So that epigenetic changes can also happen by this traditional medicine or herbal formulations. Correct. So that is what madam is telling. That is holistic approach of Ayurveda. It is not with regard to specific formulation. There is some patya is there regard of food habit, everything. So normally, if a physician correctly identify your prakriti, whether you are say Vata prakriti, Pitta prakriti, Kapha prakriti, by there is some question here by asking. But normally, what is the problem is that somebody having the fever, they already taken the paracetamol. The physician asks whether you have temperature. No. That means after consuming paracetamol, there is no temperature. So that's a wrong question answer. So it will mislead for the prakriti. Otherwise, you should mention like that. Yeah, I have the fever, but now it is suppressed after consuming the paracetamol 650 mg. Then it is fine. And many cases, the patient, physician will ask, what about your sleeping habit? Your night, your with the mobile or something. No, I don't have such an habit. I will sleep very good. It was totally lie. But the problem is the physician identified, okay, he sleep very early at night. So according to what getting the outcome of that questionnaire, they will prescribe the medicine, but it was quite different. That is why there is one word in Malayalam. Doctorodum vakilnodum kallam parayan padilla. So they should identify the proper prakriti, then it will be very accurate to any set of conditions. If it is properly prepared, that is the most important. Any other questions?
Thank you, sir. Thank you for sharing your knowledge with us. We would like to call Dr. Deepa KP to present certificate to Dr. Sulaiman City. We would like to call Dr.